Well, good morning, everybody. We're going to change the agenda a little bit. I'm going to lead off, and Dr. Gerson's going to come in by video. So, good morning. First, it's my privilege to welcome you and to honor Dr. Reza Khatib, who has now entered our building. Welcome. A neurosurgeon who has devoted his life to patients and families facing glioblastomas. His compassion and courage in the front lines of this disease are matched only by his vision of supporting the future of neuro-oncology. I would be remiss not to really tell you a little bit about the Case Comprehensive Cancer Center. I am its director. I guess I should have introduced myself. I'm Gary Schwartz. I'm director of the Cancer Center here at Case. Usually Stan comes before me. He does an introduction, so I leapfrog over him this morning. I joined Case about 2.5 years ago out of Columbia University, and 20 years at Memorial Sloan Kettering to run this program, which has been probably the most exciting thing in my life, and I've been so delighted to be here in Cleveland to lead this program. As a cancer center, it's unique. We are a consortium cancer center. Our funding is at the Cancer Center coordinates cancer research, education, and training activities for Case Western Reserve, and also the hospital systems, University Hospital and Cleveland Clinic. Therefore, we're considered comprehensive because of our unique position in Cleveland and the fact that we have high level of science, and also that we engage closely with our community. In fact, this year we just went through a big celebration last week. Every 5 or 7 years, cancer centers need to go through renewal. And when I arrived at Case 2.5 years ago, the renewal process had to be initiated. We spent a year writing the grant, 8 months preparing the grant on a site visit. We were awarded a ranking of outstanding, which is considered one of the highest in the country, putting us in the top 4% of all cancer centers. And after a bit of delay in funding, which was inevitable with the way what's going on in Washington, we did receive our award and our funding several weeks ago. So we had a big celebration last week. If you're here last week in our lobby, we had a big celebratory event with balloons and ice cream social and speeches that follow with highlights of the Cancer Center. It was great. It was one of the most exciting things I think we've done as a group, and I was delighted to be part of it. So I think, the Cancer Center, how do you define it? I guess in a good year, it's sort of like the best three for the teams in Cleveland. If you put the Browns, the Guardians, and the Cavaliers together in a good year, which we're still waiting for all those good years, combining that talent becomes really a strategy for our powerhouse, and our roster is exceptional. Each individual retains their strength, and we play from the same playbook across 3 institutions. And I think we really dominate the field of cancer medicine, and at least in Ohio, and I like to say we have emerged as a leader in the cancer medicine and cancer research across the whole country. So what does that mean for cancer research and patient care? It means that discoveries made in the labs at Case can move seamlessly into clinical trials at both the university hospitals and Cleveland Clinic. It means that for the 4 million people, that's our catchment area, in 15 counties, both Cleveland and the surrounding counties around Cuyahoga, all of our patients have access to the latest advances in cancer research, cancer medicine, and to newest therapies, both faster, quicker, and with exceptional access to these discoveries and programs. So it also means that because we coordinate our activities, we meet frequently as a group, both UH, Cleveland Clinic, and members of the Case, it prevents duplication. We can look at our scientific achievements and we can work in parallel, we can work together, we can collaborate, and brings together great resources, data, shared experience. And so we really together work in a single mind, which is and the goal of our cancer program is to prevent, treat, and cure cancer. And I think that's what we're doing here at Case. It's really with the support of Dr. Khatib and his work, and we work in neurosciences and brain tumor research that has really established us as one of the, I think, premier cancer centers in the country for work in brain tumors. Our strength in GBM and neuroscience and neuro-oncology is really extraordinary at Case, and you're going to hear a little bit more about Justin Lathia shortly. So it's his generosity that makes it possible, and it's Dr. Justin Lathia, who is actually a leader in our cancer program, molecular oncology program, a co-leader. He is the Reza Khatib, MD, Professor of Chair, endowed by Dr. Khatib. Justin is also the Scientific Director of the Rose Ella Burkhart Brain Tumor and Neuro-Oncology Center at Cleveland Clinic, and is a professor in the Department of Cancer Sciences at Cleveland Clinic Research. His laboratory is breaking new grounds in understanding how cancer stem cells interact with the tumor microenvironment, work that has already led to clinical trials in glioblastoma. Along with Dr. Lathia, Dr. Alireza Mohammed, a neurosurgeon at the Cleveland Clinic, holds another chair endowed by Dr. Khatib. Dr. Mohammadi is on the front lines of GBM care and is active in translational research involving innovative approaches in surgery and electrical activity regarding patients with GBM. Thanks to Dr. Khatib's generosity, this kind of innovative science and technology and leadership leads to continue allow us to flourish, hear a case, and institutions across the country, including UCSF and Johns Hopkins, whose distinguished faculty have joined us today as well. This symposium is both a celebration of scientific progress and a tribute to Dr. Khatib's extraordinary impact. Please join me in welcoming him, and thank you for being here today. I know it's not easy getting to Cleveland sometimes, but we're so delighted that you can come and be part of this, and we are just honored to have this symposium in your honor and for your exceptional contribution to what you've done for CASE, for Cleveland, and for the patients with brain tumors today. Thank you very much. I'm just going to lead a round of applause. So now we're going to lead into the video of Dr. Stan Gersten, who's the Dean of the School of Medicine, and he was my predecessor as Director of the Cancer Center.
Good morning and welcome. I'm very sorry that I'm unable to join you all today. I'd like to extend a sincere welcome to Dr. Reza Khatib, whose generosity and dedication to advancing research brings us together today. The Khatib shareholders within the consortium of institutions are not only researchers of the highest order, they also share Dr. Khatib's vision for improving outcomes and saving the lives of those who are stricken with devastating brain tumors. I hope you find today's presentations informative. I'm confident that sharing updates on new advances and discoveries will spark deeper connections among this cohort of researchers. Dr. Khatib, your commitment to this vital work is exceptional. I hope that today's presentations bring you a sense of pride and accomplishment by seeing that your support has put into action. And now I'd like to turn things over to Dr. Gary Schwartz, Director of the Case Comprehensive Cancer Center, who holds the Peter and Lori Weinberger Professorship in Cancer Research. The Case Comprehensive Cancer Center is one of just over 60 NCI-designated cancer centers in the country, and with Dr. Schwartz's leadership, one of the highest ranked. We partner with our consortium institutions, university hospitals, and the Cleveland Clinic, where we start with basic discovery in cancer, progress to targets, and validate the impact of blocking a pathway in study settings, move towards early phase clinical trials across diseases with clinicians at our hospitals and national consortiums, patent our discoveries, and partner with pharmaceutical companies to bring the most exciting new treatments to life. Dr. Schwartz's post at the Case Comprehensive Cancer Center follows 30 years of extensive work in early phase clinical trial development from benchtop to bedside research in gastrointestinal cancers, melanoma, and sarcoma. He is internationally recognized for his focus on early drug development and immunotherapy. Thank you, Dr. Schwartz. And once again, thank you to Dr. Khatib and to all of you coming together today in a spirit of collaboration and commitment that is vital to meeting our goal of preventing, treating, and ultimately curing cancer in brains.
Khatib, I want to wish you well and say hello here from Baltimore. I'm sorry that I can't be with you and the Khatib team, so to speak, in Cleveland. I have a family wedding that I will be at. But I'm sorry that I'm going to miss it because Cleveland is sort of where I grew up. And I love coming back to Cleveland. It's a great city. And the collection of people there that are going to be talking about the advances in brain tumor field and the treatments, much to your credit, are all gathered there today in the great city of Cleveland. So I'm sorry that I'm going to miss that. I do want to, of course, thank you for all your gifts and support of our program here at Hopkins, including the professorship that I'm so proudly holding. The progress we're making here is significant. Dr. Vadgala, our new chairman, is emphasizing clinical trials and pushing the field. It's a great opportunity, and with the resources that you're providing us, we really have a chance to make a difference. You know, with the establishment of liquid biopsy and CSF markers, we're now following or trying to start to follow patients in terms of disease progression with markers in the blood and in the spinal fluid, as well as to look at treatment responses. And we have a great opportunity with the new technology of focused ultrasound, as well as targeted drugs that are being developed and new innovations to deliver those drugs as opportunities to make progress. So thank you again so much for all of your help and assistance. It's been great getting to know you over the last few years. I've enjoyed very much my visits with you and your insight and commitment to our field. So thank you so much.
Okay, good morning everyone. I'm Justin Laffey. Let me also formally welcome you, and I'm going to shepherd us through today. So our first speaker is a colleague of mine, Dr. Jennifer Yu, who's a physician scientist at the Cleveland Clinic. She's a radiation oncologist, a practicing radiation oncologist, and she has been collaborating very closely with Dr. Ali Mohammadi, who is a Khatib chair at Cleveland Clinic, who we'll hear from later. Jennifer is really going to talk about some pioneering work involving the ability to measure electrical activity in glioblastoma. So go ahead, Jen.
Thank you, Justin. And thank you for all the support from your family to help us to move forward with some of these very exciting studies. We're really happy to share with you this work that I've been doing with Dr. Mohammadi and a large team of physicians and scientists. And working together, we can create something that's so beautiful and I think transformative for the care of our patients. So I'll talk with you today about some new frontiers in glioblastoma and specifically looking at the tumor neuronal interface. So I remember going through training and learning about cancer, and the focus was always about the cancer cells, the tumor cells, what's going awry in the tumor cells. But now we're starting to understand that tumors have a lot of interactions with other cells in the microenvironment, and that there's a lot of interactions between them, a lot of crosstalk between them. And you can see that in a glioblastoma, for example, you have a large proportion of glioma cells, but they're also interacting with immune cells, so the tumor-associated microglia and macrophages, oligodendrocytes, reactive astrocytes, neurons, neuroprogenitor cells. They're also interacting with other parts of the tumor microenvironment. And just like a village, they support each other, right? They support each other, they talk with each other, bolster each other to grow, to evade the immune system, to invade, to resist our treatments. And so we're starting now to look at some of the ways in which we might be able to understand how these interactions occur and what those consequences are. And in particular, look at these interactions between the neurons and the tumor cells and the immune cell populations. Oops, wrong way. Now, there's extensive crosstalk between tumor cells, neurons, nerves, and immune cell populations. They can occur locally with these synapses between nerves, nerves forming synapses with these cancer cells. They can occur through paracrine types of interactions, so interactions between nerves, immune cells, and cancer cells. They can occur very systemically as well. Some of our other work is looking at the role of the autonomic nervous system and how that might also promote cancer progression. Nerves can also interact with in this other paracrine type of manner here as well. And they're also cancer cell intrinsic neuronal mechanisms, whereby populations of tumor cells can co-opt neurodevelopmental signals. And we've been looking at this. I actually started looking at this concept when I was a graduate student, looking at protocadherins, and now looking at somaphorins and how they can promote tumor growth and invasion through co-option of these neurodevelopmental programs. And we also know that our treatments can have a lot of effects on the nervous system, and that can also impair the nerves, but also affect how the tumors can grow. And for today, I'd like to just focus on these interactions here, this crosstalk between nerves and tumor cells, as well as the immune cells in a local manner. So through the pioneering work of Michel Manji, Frank Winkler, and many others, we know that there's extensive crosstalk between neurons, glioma cells, and immune cells, and these can promote tumor invasion. We know that there's a formation of synapses. There's co-option of these neurodevelopmental programs, and glioma cells, they can actually integrate within these circuits, and they can function as pacemaker cells as well. And there's also beautiful evidence from Sean Harvey's groups and others that high electrical activity is associated with poor prognosis. So inspired by the work of others and this idea that neural activity can promote cancer progression, Dr. Mohammadi and I engaged in a clinical trial. This is the first of its kind prospective clinical trial, whereby we're looking at electrical activity and trying to understand the molecular determinants of glioblastoma progression. So what makes this trial so innovative is that we're combining advanced intracranial EEGs with custom subdural grids and stereotactically placed depth electrodes at and around the tumor periphery, where these interactions between tumor cells and nerve cells occur. And coupling that, we're looking at how the electrical activity can drive tumor progression, and look at these spatially mapped biopsies where these depth electrodes are placed, so that we can then understand at a very fine level the changes in the transcriptome, the changes in the epigenome that can drive these changes. And so our hypothesis is that high regional electrical activity can promote glioblastoma progression by fostering a pro-tumorigenic, pro-inflammatory microenvironment that supports glioma stem cell maintenance, invasion, and immune evasion. And we have a lot of questions that we're undertaking. So one, what is the spatial pattern of neural activity at this tumor-brain interface? 2, what are the spectral characteristics that correlate with local regional radiographic progression? 3, can we develop a electrophysiological biomarker to predict tumor progression? And this is really important because it has a lot of implications in our clinical management of patients. So if we can define at the time of surgery areas that are at high risk of progression through specific electrical signals, then that may inform areas for supramarginal resection, for which there's some data to support and improve survival for patients. And furthermore, we can tailor our radiation planning to boost the dose of radiation there, so we can achieve better tumor control. So this is one of our patients that was enrolled in this clinical trial, and this patient actually wound up progressing. So you can see in the maps over here in A that this patient has a tumor in the temporal lobe. And here are the depth electrodes. So one's placed intratumorally, and that serves as a negative control where we know that there's not much electrical activity. And then there are depth electrodes that are placed stereotactically around the enhancing tumor, so about 1 to 2 centimeters from the enhancing tumor. And then we perform these recordings. And I'm just showing you over here some of these recordings. So you can see this depth electrode in the inferior aspect of the tumor, the anterior, superior, as well as intratumorally. And what you can appreciate is that there's some activity in some of these contact points, right? But in others, there's very little activity. And as we know, in the intratumoral area, this area where it's right in the center of the tumor, there's minimal nerves over there, neurons. And as would be expected, there's little activity. And so we follow this, whereas in other areas, you can see that there's a lot of electrical activity over here. So we followed this patient. He underwent chemoradiation, and then 8 months postoperatively, he wound up progressing. And he wound up progressing in specific areas. As you can see over here, this is an MRI scan at the 8 months postoperative MRI scan, superimposed on the imaging from the time of surgery, where the original tumor is here in orange, and the depth electrodes are placed here in the inferior position, you can see in green, superior position in red, and then anterior position here in orange. And you can see the enhancing tumor right here, right, in this inferior aspect. And also superiorly, there's also tumor progression. And look, these correspond to these areas where we see high electrical activity. Conversely, in the areas here where there's no evidence of radiographic progression, we see minimal electrical activity. So this gives us some indication that there are specific signals that there are signals that we can, wrong way. There are signals that we can catch, and that high electrical activity might predict areas of future progression. So we teamed up with our neurophysiologists and the epilepsy team to look deeper into these areas where we see high levels of electrical activity and tumor progression. And you can see through the spatial weighting of these that there are some signals that are elevated in these areas where we see evidence of tumor progression. And furthermore, when we look at the power spectral densities here, these independent component analyses, you can see that in the red, in these areas where we see tumor progression, there are specific signals. You can see high beta gamma activity in these areas. And also in the blue, these tumor-associated sources also have distinct signals. So now we're starting to see that we may be able to find an electrophysiological biomarker for areas of progression. We're still early on in these analyses, and several of the other patients, or all the other patients that we've placed on this clinical trial so far are well controlled, and so we'll continue to follow them and see if when a larger cohort of patients will be able to pick out these similar types of signals in areas of progression. But so far, our patients are doing really well. Secondly, we're asking what are the activity-dependent molecular and immune determinants of progression? So a lot of studies have been done in preclinical models, but there's less known in human patients what exactly is driving these changes at the molecular level, at the genetic level, and the epigenetic level. So we teamed up with Dr. Frank Shea, his team, to try to understand what might be going on. And so we're looking at neuronal signals, neurotransmitters, the expression of synaptic proteins, growth factors as well, and how electrical activity within specific regions might promote progression through these upregulation of these neuronal signals. I'm a glioma stem cell biologist, so I'm very interested in looking at glioma stem cell state and cell state changes, so cell state plasticity, and how that might also be altered with electrical activity. And Third, we're also very interested in trying to understand the consequences of this electrical activity in the immune phenotype. So Meghana Burankayala, a PhD student in Frank's lab, is helping to do these analyses. And here's just some data from one of our patients who's undergone this, who's part of this clinical trial. You can see the tumor outlined here in red and the depth electrodes placed all around. And she performed single nucleus RNA sequencing of these tumors to get a better idea of what types of cells are in these distinct regions around the tumors, and what type of signaling might be occurring. And we're still very early on in these analyses, but what you can see is that within each biopsy region, there are a number of different cell types. Here you can see UMAPs for the whole population. And the different colors represent the different biopsy positions. And you can see the UMAPs of these different cell populations broken down by region. And then when we look at the different cell types, you can see that, of course, the largest cell population is the astrocyte population. These are malignant astrocytes, so not surprising. But you'll also see that in the periphery, there are neurons. So it's reduced in the intratumoral regions and much higher in these areas of the periphery, right, in the lateral, medial, and posterior aspects of the tumors. And we also see high enrichment of many immune cell populations, predominantly the monocyte and macrophage populations in the periphery. So this makes a lot of sense and is consistent with what we know in the literature. But what it shows you also is that across these different regions, you can see a lot of different cell types present and room for a lot of communication across these different cell types. Now, Frank's team can also look to see, to identify the tumor cell populations by looking at copy number variations. You can see that in this intratumoral region over here, where you expect to have a lot of tumor cells, and in this particular patient, he had an EGFR amplification. So we do see evidence of chromosome 7 gain, and this is where you expect to see EGFR amplification. PTEN loss is also very common in patients with glioblastoma, and so we can see loss of chromosome 10 here as well. And in the other areas of the tumors, you can see varying amounts of tumor cells. And here, just looking at the tumor malignancy cell types, you can see that the malignant population is predominantly the astrocyte population, which is what we expect. Oh, I guess some of this didn't show up, but glioblastoma can exist, tumor cells can exist in multiple cell states. These include the progenitor state populations, the oligoprogenitor cell states, neuroprogenitor cell states, and then more differentiated states like the astrocytic cell state or a mesenchymal cell state. And so Frank's team looked at the different cell states across these different regions of the biopsies. Intratumorally, not unexpectedly, we see high levels of mesenchymal type cells. These are areas in which there's a lot of tumor hypoxia, which favors the mesenchymal cell state. We see some astrocytic cell states there as well. And then more laterally, in the peripheral areas, you can see higher levels of these progenitor cells, and particularly the oligodendroglial progenitor cell populations. We can look closely. We can look within the cell signaling cell signaling pathways, and you can see large enrichment of many types of signaling pathways within the intratumoral regions, and some other signaling within the periphery. We also see a lot of cell-cell communication occurring in these different regions. Here are these circle maps that can tell you what types of cell populations might be talking with others based off of ligand receptor interactions. You can see, for example, in this anterior position, you have a lot of astrocytes that seem to be talking to the macrophage populations. And so now we can begin to understand which cells are talking with other cells and which pathways they might be using. You can see here these different signaling pathways, including cell adhesion molecules, NCAM, the JAM proteins. We see evidence of neurodevelopmental and synaptic protein expressions using neuroligands and neurexins, as well as immune cell signaling through CD22. So we're still early on in these studies. We're very excited about them, and we think that this work will have a large impact clinically. We feel that we'll be able to identify novel neurophysiological biomarkers for predicting tumor recurrence and progression. We'll learn about activity-dependent molecular mechanisms of progression. And from these insights, we may be able to develop novel therapeutic strategies to disrupt neural tumor immune interactions to help our patients. So with that, I'd like to acknowledge our team, including Dr. Ali Mohammadi, our fearless neurosurgeon who helps to do all of these studies, enroll the patients in the clinical trials, and do all the electrophysiological studies. Dr. Frank Shea, our guru in single-cell technologies, and his graduate student, and our team of clinicians, Balu, Andrew, Matthew, Jason, Juan, Rafi, and Matthew as well for all of their contributions. And I'd like to thank also the funding that you helped to provide to help us to get this clinical trial underway. And it's really exciting. We have a SPORE application in as well, so that we'll be able to learn more from the electrophysiological and molecular studies. So with that, thank you.
Thank you so much, Jen. We might have time for a quick question. Okay, I guess we'll move on. So at this point, we're going to continue the theme of neural activity. And I want to welcome Tanisha Connor, who's a graduate student with Murat Yildirim, one of our new junior faculty members. And this is a great example of people taking their talents in neuroscience and other fields and applying them to glioblastoma. So Murat's lab has been a great collaborator of the program, and Tanisha is going to share some really exciting updates today on her project.
Thank you, Dr. Lathia. Yes, as I said, my name is Tanisha Connor. I am a first-year PhD student studying biomedical engineering, and I'm working in Dr. Yildirim's lab. Dr. Yu's talk was a great talk leading into mine, which I'm also looking at linking tumor growth to neural circuit dysfunction in glioblastoma. So, somewhat like the previous talk that we just had. What's driving this work is looking at these glioma neuron interactions. And the interactions can occur in synapses that the neurons are actually making with the glioma cells, these pseudosynapses that the cells are having, as well as the electrophysiological interactions, which drives neural excitability. And all three of these lead to distant pathways that then work to promote glioma proliferation and invasion. And so, one of the papers I wanted to discuss is looking at this GBM remodeling of circuitry within the tumor, and actually looking at level regions that are highly connected and regions that are or have lower functional connectivity within the tumor area. And so what they did was they took they took the glioma cells out of areas of either the high connectivity or the low connectivity regions, and then they co-cultured those with cortical neurons. And what they found was that when they did that, the neurons then that were co-cultured with the high functional connectivity areas actually had these bursts of activity, and these very synchronized bursts were changed in basically these neurons that were co-cultured. And this was not seen in the neurons that were cultured with the low functional connectivity areas. And so what they did was they took this a step further and they implanted these cells then in mice. And what they saw was that the mice that were implanted with the cells from the high functional connectivity regions actually had a significantly shorter survival rate. And so another study actually also looked at connectivity, but what they wanted to look at was not just connectivity within the areas of the tumor, but also areas that were distant from the tumor. And so what they did was they looked at patients that had similar clinical values, and but one of the major differences were the areas of connectivity disruptions. And so what they found was that if the connectivity areas were disrupted closer to where the tumor core was, that actually these patients had better survival outcomes versus patients whose connectivity disruptions were actually in the opposite hemisphere or in these regions that were further away from the tumor core. And so they also looked and took this further and looked at how these differences in distant connectivity actually could influence recurrence. And so what they did was they looked at the connectivity preoperatively, and then once the tumor was resected, what they found was that in patients that had this distant connectivity disruptions, that they had recurrence, that the recurrence was more likely to happen in these areas actually where the disruption was occurring. And so they found that occurred in the ipsilesional recurrence as well as contralateral recurrence. And so these sorts of studies actually leads me to think of the brain as an electrical circuit. So I created this diagram here of where you can see an electrical circuit with many different components. You can see ions flowing throughout. And thinking about the circuit is if you change one part of the circuitry, what happens? So it's not just this change in the circuitry nearby that changes, but if you put something that has hyperactivity in one part, also these distant areas are going to change. And so in this example, this analogy, this is having a tumor in one part of the brain. And so that leads me to thinking about some questions such as, how does GBM progression alter local excitability versus long-range patterns of connectivity? Are system-wide circuit changes that emerge with, what are the system-wide circuit changes that emerge with tumor progression? Can tumor-driven disruptions in microcircuit recurrent motifs predict disease stage, survival, and recurrence? And how do therapeutic interventions actually influence GBM-driven circuit disruptions? And can we use this understanding to guide us for future treatment design? And so in the Yodrum lab, we are an optics lab. We're using methods such as wide field optical imaging. Here is an example of our one photon setup where we record cortical brain activity, as well as we have a camera that can record the mouse's pupil and another camera that can record the face. And so what that allows us to do is simultaneously look at brain activity, as well as how that's changing with pupil dynamics, facial changes such as the whisker movements, as well as provide visual feedback because our mice are running on an airball in a virtual reality system. And so together we can use this to look at how changes in the brain are also influencing behavioral changes as well. And so for this study, what I did was I implanted SB28 cells into the striatum in the left hemisphere. And for us, these mice live about 2 weeks with the tumor. And so one of the first things I looked at was interhemispheric connectivity. And so I looked at applied an Allen Atlas over the cortex. I looked at brain activity in one region in the left hemisphere and how well that correlated with the brain activity in the right hemisphere. And what you can see is that we start off with a pretty strong interhemispheric connectivity, and that connectivity steady deteriorates as the tumor progresses. And so that led me to wanting to look at the activity actually in itself. So what you can see is that the activity in the left hemisphere and the right hemisphere actually look pretty similar. There are a few differences, which is to be expected. But however, when you look at what these signals look like after the tumor, after the mice have the tumor, and the left hemisphere where the tumor is, you could see this, again, high burst of activity, which is hyperexcitability, as well as, but there's also changes in the hemisphere that never sees the cancer. And we see these long, prolonged peaks of activity that are kind of drawn out. And so that led me to want to look at what is driving these changes in this brain activity in the signal. And so from there, I looked at oscillations, so frequencies that are making up the signal. And what I saw was that there were no differences in any of the baselines, but in the left hemisphere where the tumor was, there was a significant decrease in the delta frequency, our lowest frequency at rest. However, when you switch to these higher frequencies like theta and alpha, we see a significant increase in that and the power ratios and the amplitude of the power ratios. And so that kind of led us to not want to just look at this from a static perspective, but want to look at more of a dynamic perspective of also how this is changing, how the information is flowing from one brain region to the next. And so that we looked at these specific recurrent patterns, these motifs that occur with brain activity. And what we see is that in baseline, we have these more predictable patterns of brain activity where the activity is flowing from one brain region to another. However, when the tumor is progressing, we see more of these very sharp global bursts of activity, either in one brain region and then it's gone, or in one hemisphere and not in the other hemisphere. And so this sort of motif work, we're still analyzing. There's a lot of data, but we want to look and see if we can use these differences in brain regions to actually predict tumor stages, tumor proliferation, and if recurrence is happening, as well as looking at these patterns with treatment and how they're actually changing. And so this kind of leads me to future directions. So I want to combine this information as well with looking at high temporal resolutions as well as high spatial resolutions. And for the high temporal resolutions, we are actually using NeuroPixel, which is a high depth electrode where we can look at various brain regions at various steps at the same time, look at neuronal firing changes as the tumor progresses, as well as local field potentials in various regions simultaneously, as well as looking at combining high spatial resolution information. And so in our lab, we are creating these 2-photon and 3-photon microscopes where we can zoom into specific areas that we're seeing these changes globally that I showed you, and then look at specific neurons and and their firing patterns, but not only their firing patterns, but how their firing patterns are changing in relation to neurons that might be close by, other cell types that might be close by, as well as the synapses that they're making with other neurons, or we can also look at the synapses with the glioma cells in a different color. So overall, I want to thank everyone in the Yildrim Lab for support on these projects, current members and prior members, as well as mentors, Dr. Yildrim, Dr. Lathia, our collaborators, and our funders. So, thank you.
Thank you so much, Tanisha. Is there any question? Time for a quick question? All right, before we move on, I do, since Dr. Schwartz is here, want to point out that this work has been initially supported by the Comprehensive Cancer Center. It's a great example of the Cancer Center bringing in folks who are not traditionally cancer investigators. So thanks for the support. Next up is going to be Alex Crane, who's an MD PhD student with Ty Miller, who's one of our new recruits. And we're going to turn our focus a little bit more to infiltrating immune cells, because that's a large part of glioblastoma therapeutic resistance as well. So take it away, Alex.
I'll introduce myself while I'm finding my, there it is. So like Dr. Lathia said, my name is Alex Crane, and I'm an MD PhD student in Tyler Miller's lab. So I want to talk a little bit today about sort of the mental model that we have of what happens when peripheral monocytes infiltrate a brain tumor. And we know that there's a couple of different consequences of that, but I want to start off by just showing you the stark difference in the amount of myeloid cells that we see in normal brain tissue as opposed to low-grade glioma and glioblastoma. So in some of these samples, we have myeloid populations that can make up to half of the cell population of the tumor. So clearly the cell population is quite important to the microenvironment progression and ultimately treatment of the tumor. So a couple of things that we know about the regulatory axis of this, and we know this from donor monocyte glioblastoma organoid co-cultures, is that if we treat these cells with interferon gamma, they take on a more inflammatory phenotype. And if we treat them with IL-1 beta, they take on a more immunosuppressive phenotype. And then additionally, it seems like inflammatory cells that remain in the tumor microenvironment for a long period of time also become immunosuppressive. And so there's sort of this idea that chronic inflammation may be leading to a lot of the immunosuppressive myeloid populations that we see in these tumors. And ultimately, this is really important because immunotherapy seems to be a very promising avenue for treating these patients. And so in order to improve efficacy of that, we need to make sure that we can modulate this immunosuppressive population effectively. And so kind of thinking about this and, where could this IL-1 beta source be coming from within the tumor microenvironment? One of the residents who's an alumni of the MSTP program, Bowen Zhao, had seen Tyler Miller's talk and had said pyroptosis was, a source of large amounts of IL-1 beta in the microenvironment. And so I just want to quickly go over, this is a very simplified schematic of what's going on, but pyroptosis essentially has two steps. The first is a priming step, and that priming step is generally going to be a cytokine or LPS from bacteria, and that upregulates all of these genes, including the inflammasome, including proforms of the proteases that are responsible for generating mature forms of IL-1 beta. And then the second step that needs to take place is that there's something in the extracellular environment that triggers all of these proteases to cleave, cleaving Gas-Dermans, forming pores in the membrane, and then also releasing large amounts of IL-1 beta. And so a big thing that I've been focusing on is trying to look for evidence of this in primary glioblastoma samples. So the first thing that I went to do is I looked at our single-cell RNA-seq dataset of 85 gliomas, and what I'm showing here is the green population are all of our myeloid cells. And so all of these genes I'm using as a signature of pyroptosis in our data set. And what we see is that our T cells and a subpopulation of our myeloid cells seem to be responsible for most of the expression of these genes. So you can think of this part as the priming aspect. So I have evidence that there's upregulation of these transcripts in this population. And then the second thing that I want to show is within this myeloid cell population, what are the population of cells that are expressing it? And so we think about myeloid cells in in these states of activity. So we have 2 activities that are immunosuppressive and 2 that are inflammatory. The systemic inflammatory activity program that we see is predominantly made up of bone marrow-derived macrophages and monocytes that infiltrate the tumor. So it looks like the majority of this signature is coming from infiltrating myeloid cells. And so the next experiment that I'm going to show you is we take tissue from the OR. We have a discarded tumor protocol, and we cut it up into little organoids and we culture them. And so one of the first things that I wanted to look at is, do I have any evidence of this in a primary tumor? And generally speaking, caspase-1 cleavage is a really strong indicator that you have inflammasome activation, and ultimately this goes on to cleave IL-1 beta in its mature form. And then additionally, we see cleavage of the gasdermin D to form an N-terminal subunit that can form a pore. And following that up, we also see that we have IL-1 beta expression in the culture. So we have all of the elements here of pyroptosis being a source of IL-1 beta release in a tumor that is, less than 2 days out of a patient. So I also wanted to do this in some of our more controlled models where we have these same tumors that I had shown before, but we culture them for multiple weeks until all of their immune populations have died in there. And I can co-culture them with donor monocytes or any sort of immune cells. And so what I wanted to see here is I wanted to see whether or not we had any evidence of infiltrating monocytes being able to undergo pyroptosis. And so during this co-culture, I take them out and I wash off all of the monocytes that aren't responsible for infiltrating. And then I have Western blot data showing that when I do this, I do see gasdermin D cleavage. And so looking at the IL-1 beta concentration, I have quite a large magnitude of IL-1 beta expression when I co-culture PBMCs with the organoid itself. So I think that there's good evidence that these infiltrating monocytes are responding to damage in the tumor microenvironment. And I think a couple questions remain. One is what cell population is responsible for priming them? And then second, looking at other forms of inflammatory cell death that could be contributing to the release of IL-1 beta as well. And so thinking about that, the way that our lab kind of thinks about myeloid cells that infiltrate is we think most of them die, and pyroptosis is a form of programmed cell death. And so it kind of seems that a lot of the cells that we know die when they infiltrate are releasing this inflammatory cytokine and contributing to the immunosuppressive phenotype of myeloid cells that do survive in the tumor microenvironment. And so with that, I want to thank Dr. Miller, Bowen, and the rest of the lab for all of their help.
Thank you so much for sharing, Alex. Any questions? Oh, good. We have a question.
Very nice talk. Quick question: the organoids are from iPSC cells, iPSCs?
No, so these are
or they are the tissue explants.
They are, so we get tumors from the OR and we dissociate them and culture them. So it's a primary patient model.
Do you have any vascularization in these organoids?
The answer is yes. So if we stain for vascularization on histology, there are, blood vessels there.
Okay. Well, thank you, Alex.
Thank you.
So next, I'm going to introduce Dr. Deanna Teague, who is a new faculty recruit of ours from Northwestern. And I want to sort of thematically move into this idea of what chemotherapy is doing, why is it working, why is it not working, and her expertise is in metabolic changes as a result of therapy. So welcome, Deanna.
Okay, thanks, Justin. So I'm a new PI at Cleveland Clinic, and I'm going to be talking to you today about TMZ-induced metabolic vulnerabilities in GBM. So really what I want to do in my lab is we know that all these patients more than likely are going to get TMZ treatment. So how can we actually look at this from like a more ecological standpoint to see if everybody's going to get this, what are actually these cells that are going through this recovery phase? We'll just do it this way. So the recovery phase, these potentially will be fundamentally different than the cells that go through the extinction phase. And so to do this, one of the projects that we've looked at is we all know that TMZ is an O6-methylguanine and a few other adducts, and then it's removed in a cysteine-dependent way with MGMT. But really, these guanines are also really important structurally for DNA secondary structures for things like G quadruplexes. And so the theme of a lot of different projects in my lab is kind of marrying what we know about neurodegenerative diseases and other things within the brain and how they're regulated and how we can superimpose this onto GBM. And so we know from neurodegenerative diseases that these G quadruplexes are really important for proper localization of different DNA damaging factors, RNA splicing factors. So one of the families that we look at is the FET family, which is FUS, EWSR1, and TAF15. So that if these G quadruplexes are deregulated, then maybe these proteins are also mislocalized and they can't do their proper functioning. And so we see this in our TMZ sensitive versus resistant lines. So these are two models that I'll be using throughout this talk that are isogenic. And then we also have T98s, which are more this intrinsic or de novo line. In all of the acquired and intrinsic resistant lines, we see the cytoplasmic staining of EWSR1 and some of these other FET family members, but it's not just an increase in abundance, as we can see at the top, and it actually is true aggregation and not just kind of clustering. So we did some super resolution microscopy where we can see in 1A and 1B that we actually see these distinct proteins, whereas in the nucleus, where we know that this can be more fluid, we see this sort of like liquid-liquid phase separation. But really what our question is and how we got into the metabolic side of this is, why is this happening? So it's really well studied for neurodegenerative diseases. You have RNA buffering, you have a lot of different kind of phenotypes that we can show in the nucleus that go between the monomers and the dimers and kind of go into these transient aggregates. But this is functionally different from the cytoplasmic aggregates that we see both in our system and the pathological ones in neurodegenerative diseases. So one of the things that we found through a lot of trial and error, which I will obviously not show you, but a lot of things that failed, but the one that actually worked that seems to be causative in our system is this 5-oxoproline seeding these protein aggregates. So this has also been seen in Alzheimer's disease, which we can see in the upper panel, where if you have this pyroglutamic acid, also called 5-oxoproline, that this can seed some of these amyloid beta aggregates. And for Alzheimer's, they're interested in glutaminyl cyclase. So this is an intraprotein version of pyroglutamic acid, but there's also another enzyme called GGCT, which can hydrolyze gamma-glutamyl cysteine, causing cysteine to be recycled and then 5-oxoproline to be upregulated. And so we can actually show kind of dependence on GGCT instead of the glutaminyl cyclase. As you can see in the green, this is staining for 5-oxoproline, whereas in our wild type cells, our TMZ sensitive cells, some of them will actually have these reservoirs of 5-oxoproline, but that with TMZ treatment, they can actually form these more secondary structure-like phenotype. And then in our resistant cells, that's all we see. So one of our hypotheses is that since we know 5-oxoproline will come back and inhibit GGCT, it wants to prevent these from being in its like usable form. Also to note that for all of our staining, we never did see true colocalization. It's only at like a tip or an end, suggested some sort of seeding. And then we can also partially reverse our aggregation phenotype as shown by the white bar with ProGA, which inhibits GGCT, but not with PQ912, which is a glutaminyl cyclase inhibitor. And so this really focused GGCT and GBM. And so we can see that it is highly expressed, is worse overall survival. We see a higher expression in our resistant lines. You overexpress it, they grow faster. You mutate it, they grow slower. But really one thing to note for trainees and anybody thinking of projects is where this really took off for us is that we looked at the localization of GGCT and it's adjacent to EGFR. And so this is something that we can see and see where with EGFR being upregulated, we also see upregulation of GGCT as well as some of the other complex 3 enzymes for the electron transport chain. So this kind of gave us the why as to why GGCT was so important, as it's not really an important enzyme in this pathway, but we think just physically its localization was important to its being upregulated. And so this also let us look into mitochondria changes, where we could see in our resistant lines, which are in the green seahorse plot, that they have a higher baseline mitochondrial rate. And then they also have this really interesting phenotype of their mitochondrial localization, where they actually have these perinuclear mitochondria. So you can see in D in the bottom that the yellow outline is the nucleus, and you see this sort of mitochondrial clustering. And it's not because there's just more of them, but that we think that it's potentially perinuclear to the mitochondria to induce this adeoxoguanine. So after we take off our TMZ treatment, you actually still have these guanine perturbations because the cells need this sort of deregulated state. And it's also not just a functional aspect. We can move the mitochondria, so it's not a trafficking issue with short low-dose nicotazole. And then for the sensitive lines, in the bottom, we can induce this perinuclear clustering with TMZ treatment. And so what we really wanted to see is, can we induce them and can we, reduce these aggregates to make sure we're fully understanding the mechanism behind this? So what we can see is if we put in our point mutations in 2 different resistant lines that's no longer able for GGCT, E98A and E98Q, these can no longer hydrolyze gamma-glutamyl-cysteine to create that 5-oxoproline byproduct. And here we see almost no aggregates in these cells. However, if we induce some sort of reactive oxygen species, which then should induce this GSH cycle, we can see that it's really a G-rich motif dependent RNA binding protein, where we can see with the clear or white bars that we can induce the cytoplasmic aggregation. So our kind of general hypothesis for this is that these cells actually need the secondary structure for proper regulation to induce cell death or to induce proper kind of like cell division. But since our resistant cells kind of like the chaotic systems, that whether they have to put the mitochondria perinuclearly to induce that adeoxoguanine, or with TMZ induction to actually change these G quadruplex structures to mislocalize and deregulate a lot of these kind of fundamental processes. And so we've also now confirmed this in patient samples. So again, we like to kind of marry the glioblastoma and neurodegenerative diseases. So we can see in primary and recurrent matched patient samples, so this is from the same patient before and after recurrence, that we also see this clinically. And so we can see that there's like EWSR1 aggregates in the bottom left, and we can also see these kind of structured staining patterns of 5-oxoproline, which mimics what we see in Alzheimer's brain compared to cognitive normal. And so what we think is kind of happening here is that in these cells, they're kind of clustering their nucleus or clustering their mitochondria perinuclearly to induce this reactive oxygen species to oxidize adeoxoguanine. And then this is going to deregulate a lot of these bindings of DNA repair and splicing factors. And that GGCT is just kind of a byproduct of this because it's close physically to EGFR. And so what we want to do now is see if we can use kind of 5-oxoproline and EWSR1 aggregates as potential biomarkers for a therapeutic that we're developing to target this metabolic deregulation. And so that's where if we have a patient sample where it's FFPE or fresh frozen, that we could test for the protein level of EWSR1 in its aggregated form, or if we have a fluid biomarker like 5-oxoproline metabolite, which is already clinically used for a very rare metabolic mutation disorder, that these could be good biomarkers for the 3rd generation compound that we're currently creating here with innovations at CCF. So with that, I'll thank the members of my brand new lab. We're almost at 2 months now. And then all my collaborators that really helped with this at Northwestern and current funding. And I will take any questions you guys have.
Okay. Thank you so much, Deanna. Any questions? Oh, okay. Dr. Schwartz has a question.
First, exceptional talk. Unbelievable. Welcome to Cleveland. Welcome to Case.
Thanks.
I'm so impressed. So you're going to block cysteine. I just want to understand the therapeutic approach and how you're going to approach it. Can you just discuss that a little more, how that is going to, I mean, how, you presented so much data. I want to sit down and chat with you sometime about this because it's extraordinary. But why do you think that blockade will be the way forward in terms of a therapeutic opportunity?
Yeah, so the reason I did not present that story is I am currently in the patent process with that drug. So basically what we found is that there's a dependency for cysteine uptake that we found from basically finding this potential biomarker. So we've been creating compounds that will specifically target redox sensitive states of cysteine that are only dependent in these TMZ resistant cells. So it's actually the compound that we're creating generally will actually strengthen potentially like neuronal signaling, but specifically target the low pH environment of the TMZ resistant cells. And so this is mostly just a biomarker for that cysteine recycling, because whenever you hydrolyze gamma-glutamyl cysteine creating 5-oxoproline, then cysteine is recycled to do other functions within the cell.
Have you ever tried blocking, looks like glutamate is a key part of this. Mm-hmm. So glutamine to glutamate, they're glutaminase inhibitors. Have you ever thought about testing that?
So we did do those. They do work great. They're not blood-brain barrier penetrant. And so these cells are also way more sensitive to arastin, so SLC7A11 transporter, which is a glutamate cysteine antiporter, whereas the sensitive cells really are unfazed. So you can treat them with arastin, that SLC7A11 inhibitor. They can find other ways, whereas these resistant cells seem to have a really large dependence on cysteine import. And then obviously a byproduct of that is the glutamate export. So then there's other kind of potential immunological aspects of that because you're exporting out a lot of glutamate. But yeah, happy to sit down and chat whenever.
Thank you, Deanna. So moving on, one of the other strengths of the Comprehensive Cancer Center is imaging. And as you know, if you can't see the tumor, you can't take it out. So Sri is representing the imaging group and is going to share some updates of their work.
Well, do you guys are aware of Murphy's Law? Everything that could go wrong goes wrong. Well, that has been my day today. So if it continues to follow me into the presentation, then don't mind me, it's the Murphy's Law. Okay. Thank you for the introduction, Dr. Lathia. Just gonna... Okay, great. Well, I am Sri Gongala. I work with Dr. Badwe as a research fellow. And today I'll be talking about the kind of clinical challenges that we see in glioblastoma and how we as a lab are trying to address them using quantitative MR imaging. Our research is supported by Siemens Healthineers. Okay, thanks to all the amazing people who have done the job for me by giving a great introduction to glioblastoma. So I'm going to skip introducing the tumor and walk straight into imaging in this field. Usually, a tumor on a glioblastoma patient looks like an enhancing ring on a post-contrast Stephen-weighted image, and this is typically how we diagnose the patient. And over the course of one's treatment, patients do undergo multiple MRI scans, such as pre- and post-surgery, as well as pre- and post-chemoradiotherapy. Despite of being one of the imaging-heavy cancers, we still have considerable limitations when it comes to accuracy in diagnosis, as well as aiding with treatment decisions. Therefore, we as a lab have come together to develop and translate imaging tools, especially quantitative imaging tools, so that we can lead this into a personalized medicine for cancer patients. I'll be talking about two projects from my lab as an example. One of them is going to be about an infiltration prediction model that we have developed using quantitative MR imaging. And the second one is understanding brain health parameters in glioblastoma outcomes. Okay. Coming to the first project, which is magnetic resonance fingerprinting, or MRF. It is a quantitative imaging tool that we use in our lab to better understand glioblastoma. It typically generates a T1 and T2 map that has true tissue relaxation times. And it also generates multiparametric tissue maps that assist in better tissue characterization and also significantly reduces the scan time. And one of the prettiest things about this is it gives T1 and T2 maps the way you see here, with a tint of red or orange and a tint of green and blue here. They look very pretty. Well, our lab has been diligently working towards bringing this tool into the world of neuro-oncology. And we have, Dr. Bardway, in her initial papers, did show the ability of MRF in differentiating different brain tumors, such as brain mets versus low and high-grade gliomas. Today, I'll be talking about how we have deployed this bad boy into prediction model. So the prediction model is a tumor infiltration model. As we have discussed before, on baseline imaging, the tumor looks with very clear margins on a T1 post-contrast image. However, as Dr. Yu mentioned, that the tumor does infiltrate beyond the tissue region here, and understanding the infiltration could be important in understanding where the reoccurrence is going to come, because reoccurrence does happen in about 95% of the patients. FLAIR imaging does give us an approximate idea of the tissue that has been affected due to tumor. However, we still cannot accurately identify the high and low infiltration regions. Therefore, and we believe, of course, understanding infiltration does aid with better radiation treatment planning, such as the CTV and GTV volumes. And again, it also aids with supramarginal resection. Thus, we have used our MRF data in constructing something like a tumor infiltration prediction model, as you see here. We use both multiparametric MRI along with MRF data to do this, and we have validated this using targeted tissue biopsies. So we use T1 weighted postcard along with FLAIR and T1 MRF map, which is with true tissue relaxation times. And we come up with this infiltration risk map that has this red and blue regions. Red indicates risk for high infiltration, whereas the blue indicates risk for low infiltration. Eventually, we have validated this using tissue biopsies, and the dots here are the regions where the surgeon has taken the tissues from. And the red dots indicate that they have found an infiltration glioma there, whereas the blue indicates that there was no infiltration glioma. As you can see, our prediction model came pretty close to predicting this. In general, our accuracy is about 80%, and our predictions are even faster than drawing a manual ROIs in order to understand the infiltration, which are usually done by a radiologist. With that, I will move on to second project, which is more my focus in this lab, which is understanding brain health in glioblastoma patients. Before we dive into the project itself, let's understand what brain health or brain reserve means. It is the ability of brain to maintain its cognitive capacity despite of undergoing age and disease-related changes. Disease-related structural changes. These structural changes could be gray and white matter volume, gyrification, cortical thickness, sulcal depth, sulcal width. The list goes on. And also, it intuitively makes sense, and also many papers have shown that increasing age is associated with decreasing brain health and thus decreases the overall performance of an individual. These have been seen to be one of the important markers in Alzheimer's disease and other neurodegenerative disorders, along with stroke literature. However, the role of brain health parameters in glioblastoma has gotten a limited attention up until now. So we are trying to address that limitation through two projects. One is a subjective analysis of brain health markers, and the second one is a quantitative analysis of brain reserve parameters. With that, let's get into the first project, which is radiological markers of brain health. These markers could include global cortical atrophy, white matter hyperintensities, perivascular spaces, lacunar infarcts. Again, the list goes on. As you can see here is an image of different degrees of global cortical atrophy and white matter hyperintensities across different patients at baseline who have glioblastoma grade 4. And as you can see here, these varying degrees do affect survival outcomes in this patient. And we have shown that in a study where we have analyzed 144 elderly glioblastoma patients. And we have seen that along with GCA and white matter hyperintensities, lacunar infarcts are also significantly associated with survival outcomes in glioblastoma patients. In this study, along with brain health markers, we have also analyzed tumor characterizations, where we have seen ependymal involvement and satellite lesions to be two of the significant parameters that are associated with survival outcomes. Trust me, this is the last slide. Well, the second project is Brain Reserve, where we are trying to quantitatively analyze the role of brain reserve parameters in survival outcomes. So we have performed morphometry analysis by segmenting gyrification, cortical thickness, sulcal depth, all that I have spoken about before on baseline glioblastoma patients. And we have analyzed about 857 IDH1 wild-type patients in collaboration with Respond Consortium. This is a consortium that's being currently maintained by University of Pennsylvania, and they have high-grade glioma patients in their data set. Well, we have seen that these brain reserve parameters act as imaging correlates for performance status and age, where we have shown that gray matter volume, gyrification, cortical thickness, and also other morphometry parameters are significantly associated with survival outcomes. We should, the future studies should definitely focus on how these brain reserve parameters can aid with treatment decisions, especially in elderly patients. With that, I would like to thank our funding support, especially my lab. That's my PI, Dr. Badwe. We are all very smiley because of her. Thank you so much. And that's me, hiding.
Okay. Thank you so much, Shri, for sharing that. Do we have any questions? Okay. Why don't we move on to the final speaker of this first session? Oh, sorry, Dr. Khatib.
I'm sorry.
Oh, sorry.
Yeah. I just have a question. Are you getting any closer for the treatment of GBM by putting all these beautiful, wonderful, very expensive research together? Please answer me.
Thank you for asking a tough question. I think the short answer is yes. And the reason I say that is because a lot of the research is fueling really novel clinical trials. And I think we are not only understanding the complexity of the disease better, but new therapies are available. This was a great example of how imaging is important. Dr. Yu provided an example about the electrical activity. And I think that I'm optimistic that we will be able to increase survival and quality of life through these measures. So as you know, it takes time, but I think we're making a lot of progress. You know, I started in this field in 2008, and from then to here, I can see really a quantum leap. So, you know, it's going to be a long road, but I think we're making really good progress. It's not soon enough. I agree.
I always hope that we have the result for the treatment of the GBM while I am still alive. Are you promising this?
I'll be very careful what I promise, but with your inspiration, obviously, we are thinking as creatively as we can. So thank you. All right, so our last speaker, let's thank Shri again. So our last speaker is Gary Gallia, who's coming to us from Hopkins, who's actually from the Khatib Brain Tumor Center and is representing the efforts at Johns Hopkins. So take it away.
That one? No. Yes. Yeah, can you just open that one? Can I see it first? Before it's on the screen. Mm-hmm. That's it. Okay. Thank you very much.
Thank you. Well, good morning, everyone. It is truly an honor for me to be here today, and I would like to thank the Case Comprehensive Cancer Center for hosting such a wonderful symposium in honor of Dr. Khatib, and I'd also like to thank Dr. Lathia for putting together such an incredible high-level program. So it's a privilege for me to be here. We heard a little bit from Dr. Weingart earlier today, and Dr. Weingart and Dr. Brem and Dr. Betagauda all wish they could be here today, and they do want to pass along their regards to Dr. Khatib and the group here. Before I start, I would like to also recognize and thank Dr. Khatib for all of the support, the incredible support that he has given for our institution and many others. I found this list of project summaries from his website, and specifically for us in Baltimore, the establishment of the Khatib Brain Tumor Research Center in 2021, the center panel here. Most recently, the Khatib Endowed Professorship earlier this year, when Dr. Weingart was the inaugural recipient for that. And in preparation for today's talk, I went back and I re-listened to that event, that beautiful event that day. And this is a quote from Dr. Khatib and his comments about his quest, and we just heard him speak directly about curing this disease. And I think this really resonates and really exemplifies why all of us are here today in this shared vision. And Dr. Khatib has also funded numerous Khatib Fellows at Hopkins. It was great to see Sina today, and all are doing really wonderful things. So I'm Gary Gallia. I'm a member of this large group of neurosurgeons at Hopkins in Baltimore. More specifically, I'm one member of this group of 12 neurosurgeons that do brain tumor surgery, the majority of whom's clinical and academic focus is on glioblastoma. And it was hard for me to think about what I can present today that would exemplify this group. So I'm going to share with you some personal experiences that I've had in my laboratory and in my practice. And I think it's a story that really highlights how we can take something that we found in the laboratory and translate it into a human clinical trial. And so this all began with a very unexpected finding. I mentioned there's a lot of brain tumor researchers. We have a very active vivarium. There's several hundred animals with tumors in them at any moment. And during this, there was a portion of time where the animals just were not growing tumors. All the experiments that had ongoing tumors, and we were taking a lot of tumors from the operating room into animals, just were not growing. And this really was puzzling for a while, and there was a very astute and bright postdoctoral fellow, Rennie Bai, who's shown here in an article about this, looked into this and with discussions with the veterinarian had determined that the animals had a pinworm infection, so they were being treated with fenbendazole. And so that was an interesting hypothesis that he had, and fenbendazole is a drug that's usually used to treat helminthic infections, pinworm infections in animals. And so this led him to look at the commercially available betazotamols, which fenbendazole is in this category, and there were 4 of them. And based on the in vitro data and that these were FDA approved, we further looked at mebendazole and albendazole. And this was the first paper looking at this. And these mebendazole clearly extended survival of this murine model. And albendazole did as well. It was at this period of time when there was a flurry of papers that came out on glioblastoma stem-like cells. That prior paper I showed you was a mouse model, and there were several. There was a flurry of these, and there's a great, very highly cited review by Dr. Lathia for people who are interested in looking at all that literature. And essentially, these glioblastoma stem-like cells better represent what a human tumor is than sort of murine cells that are grown in serum. In serum here. And so I was involved with a lot of banking with all of my cases, and we made a lot of these models and then eventually tested. These are just some examples of some of the stem-like cell lines we were able to establish. And then we went to test this in these human stem-like cells, and we found that mebendazole also significantly extended survival in this animal model. And these are, these tumors are fluorescent, and so you can look at the counts of the cells. These are control-treated animals versus at day 20 with mebendazole with a significantly decreased amount of intracranial activity. It's an easier way to assay animals than to sacrifice them. So what is mebendazole? It's an anti-helminthic agent. It's been approved in the early 1970s, and it's used for pinworm infections and for other helminthic infections. How does it work? It is a tubulin a disruptor, and this is a tubulin assay, and this is usually found in the pellet, and then when treating with mebendazole, you see that there's less polymerized tubulin in the pellet. And colchicine is a positive control, just to show that it's sort of gone. So that's the mechanism. And looking at sort of what it does in the human stem-like cells, you can see that the red is the tubulin. You can see that it really disrupts the cytoskeleton in these cells. And at that time, there was a lot of other data in the literature looking at mebendazole. These were other xenograft models in small cell lung cancer, the red showing reduction in tumor growth. This was in a melanoma model. This was in a liver metastasis model showing that animals treated with mebendazole had less liver metastasis than in control. And there were several other papers similarly had in vitro data. And there was some clinical work as well. This paper was from 2011. This patient had a liver metastasis. He actually read about mebendazole and asked his doctors, could he be treated? And they put him on mebendazole, and he had a reduction in his liver metastasis. This is another patient that had metastatic colon cancer, had liver and lung metastasis reduced with mebendazole. So this was very interesting for us. It led us to want to explore this a little bit further. Mebendazole is not one specific agent. It has three different polymorphs based on the structure, and so we looked at those different polymorphs A, B, and C, and we found that this is another animal model found that mebendazole B and C extended survival in these mouse models, and this was just looking at different commercially available forms or forms that we had manufactured. S2017 was pure polymorph C, and that sort of had the best survival in our animals. And this is a busy slide. I'm just going to highlight two things relative to intracranial tumors. One is that this had B and C had significant plasma concentrations. Mebendazole A did not get significant plasma concentrations. Probably explains some of the in vivo data. And this is a very neat experiment. So animals were implanted with tumor and then treated with mebendazole. And mebendazole is found in the side of the brain tumor in these white bars at 2, 4, and 6 hours. And it was also found in the contralateral non-tumor infected brain, showing that there's a significant amount of mebendazole that's in the brain during treatment of these animals. And so maybe just to summarize the first part of the talk here is just mebendazole we found to be efficacious in syngeneic and human glioblastoma stem-like cell lines in vitro and in vivo. I didn't show this data, but we have enhanced activity when combined with Temodar. Polymorph C is the most active of the three polymorphs, and this polymorph achieves significant intratumoral and intracranial concentrations. So at this junction, we wanted to translate this into into a clinical trial, and a lot of this credit goes to Henry Brem, who established a clinical trials group within the Department of Neurosurgery. So this trial was a single center investigator-initiated trial that was done through a surgical department. And we had a lot of questions for this study. What patient population are we going to do? Upfront, recurrent disease? What's the trial design going to look like? What dose should we use? It's been used in humans for infections, but what dose should we use for a brain tumor patient? And there were multiple challenges along the way. I'm not going to go through most of these, but mebendazole was not available for purchase. We had to have it manufactured. The FDA made us do a phase 1 study because although it has been used in humans, it was not used in patients with brain tumors before. And funding is always a challenge for us. And this was funded by 2 private foundations, one of whom at the very end decided to withdraw their funding. So there were a lot of challenges along the way. But we essentially wrote the trial to be within the standard of care. So patients, everybody in this room knows, get surgery and then concomitant Temodar and radiation, and then they start Temodar. And so mebendazole was inserted when the adjuvant Temodar starts. And it was just built into a standard treatment. Our primary objective in this was to determine the maximal tolerated dose of mebendazole in combination with Temodar. And then there were several secondary objectives in terms of safety and toxicity, plasma concentrations, and we did, even though it was a phase 1, we did look at progression-free and overall survival. So as I mentioned, this was a single institution, non-randomized phase 1 trial. It was a standard 3x3 escalation, and then there was an expansion cohort at the highest dose. These are the doses that we looked at, 25, 50, 100, and 200 mg per kg per day. Turns out to be a lot of tablets for a patient to take at these higher doses. But we wanted to escalate to the maximum dose that had been used in humans for some heightened infections. This was a paper that came out a couple years ago, and just to go through a little bit of this, 75% of patients, we included malignant gliomas, so 75% had glioblastoma, more than half of them were unmethylated tumors, and 75% of them had wild-type IDH1 on analysis. Some of our genetics were not complete. It was at a time right before all of that became standardized in our institution. And for the results, we did not have any DLTs in the first cycle, and we were able to escalate to the maximum dose. There were no severe AEs that were attributable to mebendazole, and we actually did not reach an MTD. And we did expand to 15 patients at the highest dose. There were 4 patients that did have some elevation of their LFTs at cycles 2 to 5, all which resolved with either reducing the dose or stopping drug. In terms of survival, the mean overall survival from data diagnosis was 21 months. And we did find for progression-free survival that patients that received more than 1 cycle, their median progression-free survival was 13 months. And for those that had received less than 1 cycle, it was 9.2 months. 47% of patients were alive at 2 years, and 25% of patients were alive at 3 and 4 years, and 2 patients have been on the study for over 5 and 6 years. This is the progression-free survival and the overall survival curve for that. To summarize the second part of what I wanted to talk about today, we did achieve escalation up to 200 mg per kg per dose. The treatment was very well tolerated, and the only delayed toxicity was elevated AST and ALT, and there were encouraging signs of clinical activity. So just to take a more broad view on mebendazole, this is a very active area of investigation. There's a lot of data, not only in gliomas, but in lung cancer cell lines, in medulloblastoma, in breast cancer, in melanoma, in GI cancer, so a lot of interest in this. I'm not going to go through a lot of these studies. This was a metastatic colorectal cancer a study that looked at 40 patients. Half were given Avastin, Folfox, and a placebo pill, and then the other half were given Folfox, Avastin, and mebendazole. And you can see that in terms of the partial response, the patients that had mebendazole had a much higher partial response than those treated with control. Flip side, the patients that did not have mebendazole, that had placebo, had significant more progressive disease, and there was an overall benefit in progression-free survival. So what does all this mean? In terms of future directions, we've thought a lot about trial designs, we've thought a lot about combination therapies, we've thought a lot about biomarkers, and I wanted to highlight a couple other studies relative to this, in that IDH1, a mutation, we also looked at this, Dr. Riggins looked at this specifically, and he found that mebendazole did work in the IDH1 mutants, and there was a slight improvement with radiation. This solid curve is mebendazole alone, and then the dotted line is mebendazole and radiation. So whether or not this should be preponed in treatment and given earlier with radiation comes up. And then one of the most fascinating things that I think, and I'm so happy to hear that the first two talks from Dr. Yu and from Tanisha on this neural network, in terms of, it's not just targeting the cancer cell, it's the network that has to be targeted. And these are a couple beautiful reviews, and we had a couple beautiful talks this morning about that. And so I'm going to talk a little bit about a chloride a channel and an inhibitor, bumetanide. This is a NKCC1 inhibitor. In this particular animal study, they looked at bumetanide and temodar. Bumetanide by itself had no activity, but when combined with temodar, had significant increase in survival in these animals. And so I've been working with a scientist, Dr. Bernari, about combining this with mebendazole, and he had this paper that just came out very recently looking at mebendazole and bumetanide in gliomas, and showing that the combination therapy has significantly increased amount of cell death compared to single agents, and that the mechanism is mostly via an apoptosis event. And he has a recent, this is available online, but it doesn't come up you can't find it very easily, but it is available online. This is a case report of 2 patients that were treated with him. The first one did have a high-grade glioma. They were only treated with bumetanide, and they actually did not have a response. The second patient in this study had metastatic breast cancer, and she had an anterior skull-base metastasis in a pineal region metastasis, and she had a great clinical and radiographic response after 2 months of treatment in both of these lesions. I'm going to wrap up here and just say that there's significant preclinical data on the efficacy of mebendazole in gliomas and other cancer types, which I mentioned. It is very well tolerated in patients with malignant gliomas, and we did have some early evidence of clinical activity. And I really think that we're poised for additional clinical trials, and we've been looking at a couple different options here. One is, as I mentioned, should we combine it with temodar and give it concomitant with radiotherapy, or even give it in the neoadjuvant setting to start? And then we've been looking at combining mebendazole and bumetanide to have this dual targeting. So we have this antitubulin targeting effect, and then we also have this neural network targeting effect, and to sort of maximize that. So I'd like to, this is my final slide, I'd like to just say a couple other words here. I would like to again acknowledge and thank Dr. Khatib for everything that he's done for our institution and other institutions in establishing the Khatib Research Brain Tumor Center at Hopkins, the endowed professorship in neurosurgery, and all of the fellows that you have funded for us. I'd also like to mention a lot of the folks that have done the preclinical work and the clinical translation and the clinical efforts with myself. And then I'd like to thank Dr. Brem and Dr. Betta Gowda for their vision and direction for our department. And again, thank you for the opportunity to be part of this wonderful symposium.
Randy, thank you so much, not only for attending, but sharing that really elegant story about how laboratory observations can end up in clinical trials and potentially impact patients. So we probably have time for one question. Okay, Dr. Khatib.
I heard Dr. Henry Brem now in the International Neurosurgical Conference in Los Angeles. And I heard that he's going to have a good news to tell the entire world. I am just hoping that good news is for the treatment of the GBM. Am I hoping too much? Or and I'm thinking that I should live another 100 years? Or I am going to have my 95th birthday on October 11th, and I have the good news that you have a treatment for the GBM.
Yeah, thank you, Dr. Khatib. I'm not exactly sure what surprise Dr. Brem might have for you. What I can say, and I didn't highlight for this. The clinical trials group in our group has dozens of trials that are open that are run through neurosurgery, and he is. I mean, I shared one story with you of one thing that we took from a serendipitous finding to a clinical trial that has some evidence of some disease, although it was a phase 1 trial and the goal was not to look at survival. We are very interested in doing that. Dr. Brem's career, his laboratory for decades has similar findings where, while great preclinical data, but it sort of stopped there. And I think that's often the bottleneck in terms of getting things to patients, is that a lot of good preclinical work, a lot of good animal models, and then they never kind of are tested. And I think that his focus now is to go back and to take the best data that he has shown over the decades and try to figure out how he can move that with his translation center into clinical trials. And I talked to him a couple weeks ago when I told him I was going to be coming out, and then I talked to him about that I was going to share the mebendazole story, and he was like, why is that not in a phase 2 study right now? So he is very active and motivated, and we heard from Dr. Weingart earlier. Dr. Bettigauda is the same. I mean, we have all of these great observations, but they really need to be, extended and evaluated, in patients. And I think as we understand more, and a lot of this work is, I really go back to the extraordinary symposium that is put on today. There's so much more known about GBM, and it's not just sort of one. My personal view is it's not going to be one thing that kind of treats it. I think it's going to be, targeting many different areas of that tumor. So, but maybe we will find out in October on your birthday what Dr. Brem had in store. Yeah.
First, great talk, and perseverance will pay off.
Yeah.
It takes time. Question mechanistically. It seems to prevent tubulin binding, and colchicine and this drug share some similar activities in terms of tubulin activity. With colchicine, I'm just trying to learn the mechanism. Would just colchicine, which also does the same thing on a cellular level, have a comparable effect in your tumor models? Has that ever been tried, or any mitotic spindle poison? Because we've been trying drugs in this field forever and have not had great success. It just makes me think there's something different going on with this drug that is really more than just the tubulin effect. So, could you comment on that?
Yeah, it's a great question. I think so. We have not tested vincristine in our models. We've sort of used it more as a positive control for some of the in vitro work. There are many other mechanisms of mebendazole that I actually didn't get into. There's a significant effect on VEGF and some other things that probably relate to perhaps some differences that can be seen between those two. But it's a great point. I mean, the story is a little more complex with just the tubulin effect for mebendazole. And I think people are still trying to understand the mechanism of mebendazole a little bit more. But yeah, excellent point.
Okay, there's one more question before we go to break.
Sorry, it's a very simple question. Did you see an effect did you separate your paper when you were looking at the effect of mebendazole on glioma patients, where you've looked at high-grade gliomas and low-grade gliomas separately?
Yeah, great question. That paper was only on high-grade, newly diagnosed high-grade gliomas. So it was only grade 3 and grade 4. 75, most of them were GBM, and then there were several anaplastic astrocytomas, which we included because the primary endpoint of that study was a safety study and not an efficacy study. Yeah.
Okay.
Yeah.
Well, Gary, thank you again. We really appreciate you sharing the story. I know we're running a little bit behind. The good news is we still have time on the back end, so maybe we can take a 10-minute break. There's refreshments outside. If you're not familiar with this building, go around the corner and out to my left. That's where the restrooms are.
And next.
Do you have an adapter? An HDMI adapter?
13 of those are enclosed, yes. I tried to put in your code yesterday, and I was literally almost done. I have one more thing that I have to submit because they all go in like line item and website one down. So I'm not sure if any of them say if it did, I can finish it up today. If not, then I'll get it done Monday. Thank you so much. Of course. That makes sense. So that's if you
Okay, just want to let you know we're going to start here in maybe a little bit less than 5 minutes. So if you still need to go to the bathroom or get a drink, feel free to go ahead and do so. Okay, welcome back. Before we get started with the second part of the program, we wanted to sort of share some remarks from Dr. Jean Barnett, who's the director of the Rose Ella Burkhart Brain Tumor and Neuro-Oncology Center at the Cleveland Clinic. Unfortunately, he's away and he can't make it, but he wanted both Dr. Mohammadi and I to talk briefly about the program. We have a very active clinical service, which Dr. Mohammadi will talk about, but our research program is also quite strong. We have a lot of innovative clinical trials. We have plenty of examples where laboratory discoveries have made it into early phase clinical trials, and we are active in the community at pioneering things like window of opportunity trials where we give patients the drug and then folks like Dr. Mohammadi take out the drug, and that allows us to study the response in biochemistry in patients. And Doctor, you mentioned it earlier, but we are at the very late stages of submitting a SPORE application. So it's a specialized program of research excellence. And folks like UCSF have had a SPORE for years, and ours is really focused on brain tumors, as is theirs. But I think it's really a testament to the research investment the Brain Tumor Center has made. So I'll turn it over to Dr. Mohammadi to briefly talk about the clinical operations.
All right. Thank you very much, Justin, and thanks, Dr. Khatib, for coming to Cleveland finally. So so in the Brain Tumor Center, actually, Dr. Jim Barnett was the one who thought about just putting together this Brain Tumor Center, and the idea was to have all the subspecialties that they are involved with brain tumor patients to be in one department. The way that it works, we are all in the same center. Actually, we are in the cancer center, and the neuro-oncologists, us, the radiation oncologists, we are like see patients next to each other, so the patient doesn't have to go from this building to the other one, and it's very patient-centric. For the like post-op, we see patients all at the same day, and most of the time patient stays the same room, and we go just one by one. So, like in the Cleveland, we have, that in Florida we have like the branches, and in Abu Dhabi, London, and other places. But in the Northeast Ohio, the Brain Tumor Center has like 8 neurosurgeons in the main campus and 2 in satellites. We have 7 great like scientists like Andrew, neuro-oncologists, and we have our own like endocrinologists for the pituitary the cases, and there are people from other departments that they have joint appointment with us, like 6 radiation oncologists, about 10 neuroradiologists. We have our own psychologists, and we are work like hand to hand all together. We have like 2 brain tumor boards and 1 spine tumor board like every week, and a lot of time, like Andrew or others, they are seeing a patient, they call one of us that the patient needs surgery the same day. We just go to the same room and see the patient. So in terms of volume, like I would say about 800 surgeries and more than 1,000 gamma knife radiosurgery a year. That's the care that we provide to the patients. So, with all that, we go.
Okay, well, thanks so much for the clinical overview as well. So I want to get started and invite our colleague from UCSF, Philip. I'm going to try to pronounce your last name. So, but I appreciate you sparing me then. So, go ahead, take it away.
Of course. So this is very special for me. I will talk. Actually, I feel like I'm the dumbest person in the room, which is a great place to be. And I consider myself entertainment here because I'm the only one who's not going to talk about glioblastomas. So, Dr. Khatib, I'm not going to solve the glioblastoma problem today. I will talk a little bit about, I think, an even bigger problem, the meningioma problem. So, I have no disclosures. However, I should tell you a little story. Dr. Khatib and I met for the first time in almost exactly 15 years ago. I think it was the 14th of October 2010, and I was the Khatib professor at UCSF. Back then, I was the program director at the University of Cincinnati. I used to live in Ohio. This is my backyard. And Andy Parsa, who was very dear to both me and Dr. Khatib, had my position at UCSF. We were very close in many ways. He and I started our careers together in similar fields. And Andy ended up at UCSF from Columbia, and I ended up from UCSF to Harvard and then to the University of Cincinnati. And it was a great ride we had. I remember this, from the Four Seasons over to the hospital to give the talk. As bad fate would have it, Andy passed away young. After he left UCSF, however, to go lead the program at Northwestern, I got the call to come and replace him, and non-expectedly, I ended up back at UCSF where I had trained, and I ended up taking over the Khatib Endowed Chair. And I will share with you a little bit of what we have worked over time. Now, much of this, the real disclosures, much of this is team science in many ways, and I'm just a little bit of it. There's a lot of people who will be thanked at the end. And this is an ongoing story. And as I grow older, I think I share with Dr. Khatib's frustration that we have not gotten further than we have. Truth be told, 20 years ago when I was training, melanoma in the brain was a death sentence. Primary CNS lymphoma was a death sentence. And now we consider these things almost chronic diseases. So the truth is, we have made huge leaps in this. And at UCSF, I can tell you, and in the recent political climate, it has been really difficult. We have over $800 million of NIH funding, UCSF in total, and the Department of Neurosurgery has over $30 million from NIH out of about $100 million that we get in, and it's philanthropy that now is really bridging us over this difficult time of having budget cuts and delays in funding, and you have done an amazing job. You have done an amazing job. I can tell you that now with gray hair, that much of what we've made in progress would not have been possible without this. You see here the Weil Center, Sandy Weil, another big philanthropist who has endowed us a lot of money as well. But it is from this that we go forward. And I hope that we keep on pushing the envelope as much. Why petroclival meningiomas? Well, because I thought you guys heard enough about glioblastomas today. And also because what I do directing the Skull Base Center at UCSF is mostly, is a big part of it, is meningiomas. And meningiomas, for many of you who I take it are not neurosurgeons, and I'm glad to see this kind of mixed group, is the most common tumor that grows in the brain inside the skull. Petroclival, being a location anatomically, is the hardest tumors really to have. It's one of those things that even when I hear about it, I get this like negative feeling because they never really end well. These are not easy tumors. And if you can imagine this, these images here are basically a skull looked up from the top down, and this doesn't show. Is there a pointer? Or, yeah. Oh, no, that's the other pointer. So this is the center of the skull, the sphenoid bone, and this is the origin of the tumors along this little ridge right here, the petroclival fissure. And this is the trigeminal nerve on the one side, the trigeminal nerve on the other side, the sixth cranial nerve here, seventh and eighth cranial nerve. So this is basically the epicenter of these tumors. And why is this important? And this is the pituitary gland, the carotid arteries. Why it's important is because as it does that, it pushes all the cranial nerves to the side, meaning to the location where we're coming from. So we have to come through a corridor of all these cranial nerves being compressed. It pushes most of the arteries that go to this part here, which is the brain stem, also out into the field. So the tumor is hiding behind all of the tough things that are very important and the things that feed the brain stem. I say this as an introduction because this is by far the most difficult tumor to take out of the brain. And as opposed to how many studies there are out on glioblastomas or on plenty of other meningiomas, these 12 studies are the studies that exist in the literature over the past 30 years. And none of them have really great outcomes. In fact, they have mortality rates defined. Most brain tumors anymore do not have defined mortality rates. This does. And the rates of cranial nerve deficits are high compared to anything else. And the rates of gross total resection are low. So we're not really curing these patients, and we're hurting them. Now, we decided also, and I'll share with you now our data, but I will show you only a little bit of data on two of these studies, and these are for you guys who may not be neurosurgeons, two of the most important technical neurosurgeons in the century, Taka Fukushima, 140 patients, and you see here one mortality, 8%, 7% stroke rates with hemiparesis or monoparesis, and 20-some% cranial nerve dysfunction. And by the way, these are people, Fukushima and the next one I'll show you, are people who don't believe in anything but taking the whole tumor out, and even when these people tried, couldn't. And this is Sheikh, another very pillar of skull-base surgery, 150 tumors. These are the big studies. And again, you see gross total resection 30%. You almost never say these people's name in anything but gross total resection, but you see here they just can't do it, and morbidity high as well, and recurrence rates. Finally, over this 30-year period, radiosurgery, well, many different machines has come into play. We are also a very big radiosurgery center in San Francisco, and with both the Gamma knife and Cyber knife and other knives. And you see that in this time period, 170 patients from Pittsburgh, Doug published this, and you'll see that for bigger tumors, which are most of these, the control rate, even with radiosurgery, is not good. So you have really no good option here. And this, by the way, is the tumor, the introduction to our main actor of this, the petroclavial meningioma. As you can see, it's kind of hidden by brain on all directions, so it's in the center of the skull and along the skull base. Now, in our study, we had 76 patients, and what you see is a variety of different approaches to try to get to it. This is, again, the skull. Remember, we talked about this is where they arise from. The majority of these tumors will be taken out through a transpetrous approach. This is the petrous bone here, and we drill some of that bone, the bone around the hearing apparatus, in order to even get in. And even if you do all that, then you still have to deal with all the nerves and the arteries that supply the brainstem or come out of the brainstem. Our data, median follow-up, a little bit over 4 years. Many of these patients presented with cranial nerve deficits before, and usually it's either double vision or sensation changes in their face. And the median tumor volume is 24, 23 centimeters cubed, so big. Most of these are big. In that area, things grow, and most meningiomas grow slowly. Most of them grade 1, about 10% of them not. So when you see, again, for us also, the extent of resection, the great majority of this, and even if you go back on all these studies, if you really study them with recent imaging, you'll see that basically all of them end up being subtotal resections. We leave tumor in the cavernous sinus along the skull base, etc. Average time, 10 hours, and these were done. A few were actually Andy's cases, and mostly mine and Mike McDermott's, which were not exactly new kids on the block, but 10 hours of surgery for these tumors an average. Most of them receive radiation afterwards. Many of them progress. Symptomatic progression, 62% of the patients after treatment. Overall complications, and this is I think the most important slide of all of this. Almost 30% of people with complications, and 1 in 10 will have a stroke. And these strokes in that area are serious. This is not something we see on a scan. This is you'll never walk again, you'll never move again, you'll never talk again, and sometimes you'll never wake up. I have gotten to, based on all of our data, I have gotten to tell every patient I see with this that I know I'm going to hurt you somehow. I don't know how. It could be that you just have double vision, but it could be that you're not going to move your side. And I think this is truly informed consent of this. So there's nothing really great in the outcomes. When you look at all the cranial nerves, you see here the majority of these are either related to the trigeminal nerve dysfunction or to hearing and facial nerve dysfunction. And up to 47% of them, 48% of them by the last follow-up 4 years plus later, meaning after everything is healed and done, they have a new neurologic deficit that affects them. And when we looked at the multivariate analysis of this, really only BMI showed a significant risk for complication postoperatively, mostly because these are such big procedures that complications beget complications, and the BMI can be a problem with healing. So what can we do for this? Well, we can do a lot, and this is kind of the technical thing. Post-op, pre-op, we can take them out. And this little bit stays in the cavernous sinus, and then we can treat that with radiosurgery. And you see the brainstem without a stroke, and I wish every single one of the scans was like that. This was a great one, and we usually show the great ones. Not everything is like that. When we look at the surgical approach selection, the majority of these fall into this group right here, not as critical for you non-surgeons, but basically the point away from this is that these are big approaches. These tumors extend in multiple compartments of the skull and the skull base, and so we actually have to, this approach by itself takes 3 or 4 hours to do. And when you look at this, just a little peek, this is the brainstem right here, the surface of the brainstem. This is the tumor debulked, and now I'm dissecting this tumor away from the surface of the brainstem. And this is, unfortunately, the anterior brainstem, the ventrolateral brainstem, which is where the motor fibers are. You go through this little pia here, and you will take somebody's function away, including all of these little blood vessels that feed the perforators that come from the basilar artery, which is up in front here. So this is the limiting factor in general, is how stuck this plane is of the tumor and what other vessels and nerves are within it. So, and saying this, just so that you see, this is the DTI imaging, which shows us the motor fibers here. You see, again, this is a different type of tumor and a different patient, but the same type of tumor. The motor fibers just hugging the top of this tumor being displaced posteriorly and superiorly. And this is why I show this image, because we took the great majority of the tumor, but this tumor at the very front is stuck there and it's not really possible to remove. And in the other one, you saw there was a small piece behind. This is a larger piece. And when you look at the overall rate, you see that gross total resection is only 6% of the patients. So 70% will receive another treatment. So if it is radiosurgery, sure. If it is any type of radiation, sure. But even out of those, a significant number of them progresses. So then what do we do for these patients? And this is where sort of all this support for research comes in. This is the story of great technical ability, and it's not just mine, but this is now the 13th study of this, and we can do, this is almost as well as we can do, no matter how much we try. I've done now over 7,000 brain tumors, and I can tell you this doesn't get much better, not because I'm good, but not because nobody else has been able to show that we can do better. So there is a piece of the puzzle that we were missing for a very long time in benign, quote-unquote, tumors. Tumors meaning that are not malignancies like glioblastoma. And that piece was medical therapies of any sort, targeted therapies. We used to only have surgery and radiosurgery for it. Now this is an old resident of mine from Harvard, Ian Dunn, in his lab. The research that have shown several years now back about meningiomas that don't have the NF2 mutation, which is the great majority of meningiomas, may have other mutations, in this case SMO and AKT1, but also a number of different ones that may drive it. And this was sort of the beginning of starting to think about meningiomas as tumors that are targetable. When I was training back 20 years ago, there was a couple of chemotherapies that people had for meningioma. Neither worked, and neither was targeted. And because of papers like this, we started thinking about that. Now, Michel Calamaridis, a good friend who is the chair at one of the Paris departments of neurosurgery in France, did this great study where they showed specifically for anterior skull-based meningiomas, which this one is not, that GAB1 is a very significant predictor of recurrence. Now, we know from the work that Ken Aldapi and person who was our neuropathologist at UCSF then went up to work in Toronto with Zade and then now at the NIH, we know that meningiomas, the recurrence pattern of meningiomas is dependent on whether you take them all out or not, whether they are recurrent tumors to begin with or not, but also whether they are along the skull base or not the skull base. And in general, skull-based tumors seem to be different phenotypically even. And why is that the case? Well, we don't know exactly, but studies like this from Murat's lab at Yale show that specific mutations may be specific to locations. So SMO can be specific to the anterior skull base, that the central skull base may be AKT1, etc. And this is something that will require a lot more work going forward to understand which ones of these mutations or epigenetic changes would be important. And I'll conclude with talking about the FOXM1 story, which, for the sake of time, I will not go into the great details, but this was a great finding from a lab funded partly with the funds that we have from the Khatib Endowed Chair, that basically, the long story short, showed that FOXM1 can really show us as a transcription factor, can really identify aggressive meningiomas. In fact, FOXM1, both genetic and epigenetic changes, can identify aggressive meningiomas better than WHO grading can and better than MIB count can. So we are now seeing the world where genetic and epigenetic descriptions of tumors have significant clinical implications. That has led to now 4 clinical trials that we have on meningioma from 0 out of the 65 clinical trials we have for all brain tumors, almost all of them glioblastoma. And I would say, I would leave you with this, that the optimal surgical goal is still elusive. However, we are getting to the peak of our abilities, that functional preservation is becoming more and more important, that tumors do grow no matter what we do to them, that there is molecular subtypes now we're identifying, and that translational trials are becoming now the norm, and how we will be treating these. And I will leave you with these words that that Harvey Cushing had said, that all meningiomas can be operated on, but not all should be operated on. And that should be our challenge into the future, to try to figure out different ways of being able to treat this most common tumor in the brain. And with that, this is work for many people, collaborators that I have, and thank you for the invite. Thank you to Tamara and Carolyn for helping out. Thank you to Justin for putting this all together. It's way overdue. And thank you to you, Dr. Khatib, for all the support you've given us over the years, and you're planning for more, for another 100 years, because you're going to wait for that GBM cure, right?
Okay.
Thank you.
So if there, maybe we'll move on just because we're a little tight on time. It's my honor to now introduce Sina Ahmadi, who's a Khatib Fellow who I think represents you, Dr. Khatib, in a way where he started at Hopkins and now he's here in Cleveland doing his PhD. And he's going to share some clinical updates on long-term GBM survivors.
Hello, everyone. Thank you, Dr. Khatib, for coming to Cleveland, and thank you for all your support. My name is Sina Ahmadi. I'm a Khatib Research Fellow and a PhD student at Cleveland Clinic. I'm presenting on behalf of Dr. Hamid Etemad Rezai, who was the chair of neurosurgery at Mashhad University in Iran, where I also did my neurosurgery residency. Today, I will be talking about long-term survival in glioblastoma patients. As you know, the median survival after standard therapy, which includes safe maximal resection, radiation, and adjuvant temozolomide chemotherapy, remains a dismal 12 to 15 months. And the first quarter of the second year, fifth quarter post-diagnosis, is considered to be the peak incidence of mortality. But around 8% of patients are expected to survive at least 2.5 years. And about 2% of young adults are alive at 10 years. So the result of this paper indicate that the long-term survivors are patients who survive at least 2.5 years post-diagnosis. Another definition in another article, they defined long-term survival as an overall survival equal to or longer than 3 years from the date of surgery. And they defined another group, the short-term survivor, with an overall survival of less than 12 months. So as I mentioned, one paper showed 2.5 years, another 3 years. So there is no standard definition of a long-term survival. And studies are using varying time periods to be defined patients considered to be long-term survival. We have series of long-term survival patients, and I want to present 3 cases that had more than 72 months, 6 years overall survival, and 2 patients are still alive with good functional status. This overall survival is more than twice the suggested threshold that I talked about for long-term survival in GBM patients. Case 1 was a 20-year-old female with left frontal mass was extended to corpus callosum with overall survival of 7 years. Two other cases were 14-year-old female and 31-year-old male that I'm gonna show you these 2 cases with details. All patients received radiotherapy after surgery with concomitant chemotherapy with temozolomide. This 14-year-old female was admitted in 2016 with a headache and signs and symptoms of raised intracranial pressure. As you see in T1 with contrast MRI, left frontal parietal mass, irregular peripheral enhancing and central necrosis, plus surrounding vasogenic edema with mass effect on the left lateral ventricle. As you see, MRI is characteristic of GBM, and in post-operative MRI 8 years later, in 2024, there is no enhancement in the surgical bed and no mass effect on the lateral ventricle. Pathology was reviewed and reported by a neuropathologist that was GBM. Another case, 31-year-old male was admitted in 2017 with a headache and seizure. In MRI in T1 with contrast, you can see a left temporal mass with heterogeneous enhancement with less necrosis and less surrounding vasogenic edema than the previous case with minimal mass effect. And the post-operative MRI and also MRI, this is post-operative MRI and this is MRI 7 years later, showed no enhancement and no recurrence or enhancement. This patient was not only lucky because of his GBM, but also he won green card lottery 2 years ago. Good prognostic factors for long-term survival are divided into clinical and molecular factors. Clinical factors like younger age, better baseline performance status, near or gross total resection, longer pretreatment duration of symptoms, and molecular factors like lower Ki67 labeling, MGMT methylation, IDH mutation, and lack of p53 overexpression. And levels of biomarkers of systemic inflammation were significantly different between the long-term survival and non-long-term. Something like higher pretreatment levels of absolute neutrophil count, neutrophil lymphocyte ratio, monocyte lymphocyte ratio, derived neutrophil lymphocyte ratio, lower levels of absolute eosinophil count that are related to bad prognostic factors. So survival benchmarks in glioblastoma have been evolving since more cases are now diagnosed and treatments are improving, and as rare but significant long-term survivors are identified, and more studies are needed to better understand these long-term survivors. We presented this talk to emphasize that identifying differences between long-term and non-long-term survivors can inform future prognostic models and may also guide treatment-related decisions. Finally, according to the increasing number of patients with long-term survival, whose survival exceeds the conventional definition of long-term survival, we propose the term extended long-term survival to describe patients who live more than 5 years after diagnosis. So this is not the answer of your question, but in clinic we see patients with extended long-term survival and it shows the improvement of treatment. Thank you.
Thank you, Sim. We'll move on now. So it's my pleasure to introduce Fatima Khan, who is working with Pei Wen Chen, who's been one of our recent recruits. I shouldn't say recent, it's over a year now.
1 year, yeah.
They're celebrating their 1-year anniversary in Cleveland, or just passed. And their work is very focused on the tumor microenvironment. So Fatima, thanks so much for joining us.
Thank you, Dr. Justin. Good morning, everyone, and thank you for the opportunity to present at Symposium. My name is Fatima Khan, and I'm working with Pei Wen Chen Lab in Cleveland Clinic. So today I'll be sharing our work on how lactate-driven STAT3 signaling shapes the glioblastoma microenvironment. So in our recent work, we discovered that abundant macrophage infiltration and altered tumor metabolism are tightly linked in GBM. We show that LDHA-driven glycolysis activates ERK, YAP, and STAT3 signaling and recruits these chemokines like CCL2, CCL7, and these macrophages take up these cytokines, and then it reinforce the tumor glycolysis through LDHA-containing extra vesicles. So this metabolic symbiosis sustained tumor progression and inhibiting LDHA disrupted both macrophage infiltration and tumor growth. So the unresolved question, we were thinking like, how does this lactate can derive this regulate the TAM function through like metabolite or versus epigenetics. So what happened inside the macrophages? So we were always thinking about some other pathway which can regulate this signaling. So the key hypothesis of my current project is based on the tumor-derived lactate induced histone lyse 18 in macrophages, and this epigenetic modification leads to STAT3 activation and the transcriptional reprogramming, which together drives immunosuppressive TAMS function. To test this hypothesis, we structure AIM 1 and AIM 2 and 3. So AIM 1, we are defining the LDHA lactate axis controlling the TAM polarization and activation of STAT3. AIM 2, we wanted to test whether the lyse 18 is enriched in STAT3 and promoter, and to examine how this modification impact the downstream CD8 T cells. Then AIM 3, we wanted to evaluate the in vivo impact using the LDHA inhibitor. We have published one of the inhibitor we found after the screening, and we did some therapy with immune checkpoints. So we directly tested how LDHA loss in macrophages influences their polarization. So we treated the wild type and LDHA knockout mice with lactate and tumor conditioned media, and we see a strike increase in the polarization of these macrophages. However, in contrast, LDHA knockout BMDM's treatment with lactate didn't show that much of upregulation compared to the lactate and conditioned media treatment. Then next, we also did some arginase level in like transcription levels using the arginase positive genes. We found a similar pattern there. Then to complement our genetic data, we tested whether this pharmacologically drug that FX11, it is commercially available inhibitor of lactate, LDHA. So we use this FX11 and to see the suppression, and we surprisingly see the similar effect in the macrophages. The polarization was increased with the lactate, whereas FX rescue that polarization. And among dozen of compounds, we screen a molecule like small molecules, and we screened dozen of compounds, and we found similar asteripentol and some other drug reduces the arginase level. Then we did side-by-side validation of this drug called steripental. We treated with lactate and conditioned media. We saw the polarization was decreased after the treatment, and also the transcription level decreases. Next, we moved on to RNA-seq. We isolated the LDHA knockout BMDM and control. We sent for the sequencing. We found that IL-6 JAK-STAT hallmark pathway is significantly suppressed in LDHA deficient mice, as shown in enrichment plot. To validate this finding, we examined the STAT3 activation directly, and lactate treatment increases the phosphorylation. Then we moved on, to see which mechanism it's like regulating this. So we already know there is one concept comes from landmark study Nature in 2019, where the lactate was shown to directly modify the lyse-18 in histone that was found to be one of the most abundant and functional relevant mark. So this modification linked to glycolytic metabolism to gene activation, so particularly in macrophages. So we explored some chip PCR assay. We found that similar that lyse-18 was upregulated with the lactate treatment and as well as the tumor conditioned media treatment, and this this expression was depleted by the steripental treatment. However, with the lactate and conditioned media treatment, we see a slight rescue back, significant rescue back in there. Then next we moved on to see, how the lactate drives the STAT3. Now we can see in the Western blot, these global pan-lactylation, if we see, it also increases, and the lyse-18 with STAT3 expression was also significantly increased, whereas it was depleted with the knockdown LDHA BMDM treated with and without lactate and FX treatment. Then next we examined whether functional consequences. So we isolated the BMDMs and T cells, and then we co-culture these T cell and BMDMs to see whether this conditioned media lactate can really suppress the activation of T cells. We found in CD869 positive and interferon gamma, where we see the conditioned media lactate was surprisingly suppressing the T cell function, and it was rescued back with the LDHA knockout BMDM. We also checked some viability of T cells with different conditions, and we found a similar result here as well. Then we perform a cytotoxicity killing assay, which is we use effector and target ratio. With the different target ratio, we found the T cell mediated cytotoxicity was suppressed with this target ratio, and then it was relieved by LDHA knockout BMDM with and without treatment. Now to test this in therapeutically, this is all we are just doing some preliminary experiments. So we wanted to go further and see whether this drug is really effective. So we did some therapies in two different models, CT2A and stem cells model. And then we found with steripental, we can see some modest slight increase in the survival, whereas when we did the together with the PD-1, it's surprisingly we saw the survival effect was better than the single treatment. Then we did, this is ongoing right now, so we injected the LDHA knockout tumor cells to the knockout mice, and now this is so far going on very well. Then from CD2A tumor model, we isolated the tumor tissue and spleen. We checked the polarization of macrophages in the tumor tissue. We found that the macrophage was completely polarization was stopped compared to the wild type group or the just single treatment with PD-1 and steady pentol. And also we isolated the spleen and T cells from there to see how the T cell is functioning after the treatment. We saw a significant elevation of CD8 T cells and also the activation in the double treatment group, and the exhaustion was relief with the double treatment group. So now, just to summarize this whole study, so we know the GBM cells releases LDHA, which goes and binds, interact with the macrophages and increases the STAT3 and histone lectylation, which directly hampers the CD8 T cells and dysfunctions, which leads to tumor progression. In closing, I would like to thank Dr. Pivenchen, my wonderful colleagues, and all the collaborators for their support. I look forward to your questions and discussion. Thank you so much.
Okay. Thanks, Fatima. That was very interesting. Any questions? Maybe time for one? No? Okay. We'll move on then. So, keeping with the theme of metabolism, it's a pleasure to introduce Andras Ponti, who is a joint PhD student between our group and Chris Hines' group, who mainly studies aging, and will tell us a little bit about a new metabolic pathway that he's been working on.
All right. Well, first of all, thank you to the Comprehensive Cancer Center and to Justin for these kind of opportunities for trainees to present their work. And thank you also to Dr. Khatib for supporting trainees and science here in Cleveland. So it's exciting for me to be able to share updates on my project, where we're looking at this pathway that is involved in sulfide oxidation and how it impacts metabolism and growth in glioblastoma. So this project kind of began as an offshoot from a collaborative effort between the labs of Dr. Hine and Dr. Lathia that was led by Dan Silver and Nazmin Beathy. So they were focused on understanding how a high-fat diet impact tumor progression in glioblastoma. What they had found was that in a high-fat diet, there was a reduction in this gaseous signaling molecule being produced called hydrogen sulfide, similar to other gaseous molecules such as nitric oxide or carbon dioxide. This decrease in hydrogen sulfide corresponded with a decrease in overall survival in a model of glioblastoma. And this was able to be reversed when mice implanted with a glioblastoma cell line were treated with exogenous hydrogen sulfide. As you can see that there was a decrease in tumor volume over time. Being a gaseous signaling molecule, it's a little bit difficult to treat patients with exogenous hydrogen sulfide. So what we instead wanted to do was look at endogenous pathways for producing hydrogen sulfide. And so that pathway is the transulfuration pathway. This pathway begins with methionine entering the transulfuration pathway as homocysteine. Homocysteine can then be acted upon by cystathionine beta synthase to generate cystathionine. And then cystathionine can be further metabolized by an enzyme called cystathionine gamma lyase to generate cysteine, which can go on to be used as a building block for proteins or for generating the redox molecule glutathione. But what we are particularly interested in is the abundant production of hydrogen sulfide at both steps in this reaction. So utilizing two available genetic models that we have in the lab, a cystathionine gamma lyase overexpressing transgenic mouse, as well as a cystathionine gamma lyase knockout mouse, we implanted mice with a murine model of glioblastoma, and to our surprise, we actually saw that the black line, the cystathionine gamma lyase transgenic mice that have increased ability to produce hydrogen sulfide actually were dying faster than the cystathionine gamma lyase knockout mice that can't produce hydrogen sulfide. So this was a little bit of a head scratcher to us. So then we looked at some other enzymes that are actually responsible for oxidizing hydrogen sulfide to eliminate this gaseous molecule. And so what we saw was that when glioblastoma is in the presence of hydrogen sulfide, we have a rapid and robust increase in the enzymes that are involved in oxidizing hydrogen sulfide. So this pathway is known as the sulfide oxidation pathway. And so it is a 4-step pathway beginning with sulfide quinone oxidoreductase, which breaks down hydrogen sulfide and passes the sulfur along to glutathione, generating glutathione persulfide. And the electrons are passed on to coenzyme Q, which enter the electron transport chain to be used to produce ATP. The glutathione persulfide is then further oxidized by ETHE1 or sulfur deoxygenase, which generates sulfite and again generates glutathione. And at this point, there's a fork in the pathway where sulfite can be further oxidized by TSD or rhodinase to generate thiosulfate, and then sulfite oxidase can generate sulfate. So as you can see, this pathway is involved in a lot of different functions. So it can oxidize hydrogen sulfide to eliminate this gaseous molecule. It can be used to generate this abundant list of metabolic intermediates. It also can be used to regulate this protein translational modification on cysteines known as protein persulfidation. And then it can also be used to generate ATP, and it can also regulate redox homeostasis. So as we saw that sulfide oxidation is increased in glioblastoma models, we wanted to first look at what happens in patient transcriptional data. And so we see that there is an increase in expression of sulfide quinone oxidoreductase, sulfur deoxygenase, and a modest increase that's not significant of the 2 other enzymes. Additionally, we can see that expression of SQOR and ETHE1 increases with increasing tumor grade. So then, as it looks like sulfide oxidation is being upregulated in glioblastoma, and it can help protect against hydrogen sulfide, the first thing I wanted to do was see what happens when we implant mice with overexpression, cell lines that are overexpressing either SQOR, ETHE1, or the combination of both. And so what we saw here was that whether we overexpress and implant mice with just the cell lines overexpressing SQR in the red line or ETHE1, the blue line, or the combination, we do see that there is an increase in tumor formation and disease progression, resulting in shorter overall survival times. But so then beyond just seeing does sulfide oxidation increase tumor growth, we want to know, is this a potentially targetable pathway? So then what I went ahead and did was, utilizing lentivirus again, I generated additional stable cell lines where this time we knocked down SQOR, and you can see that we have good knockdown. So when we implant mice again, here this time what we can see is that relative to the controls, there is a robust survival extension with knockdown of SQOR. So this is potentially indicating that sulfide oxidation, it both can accelerate disease progression, but it is also something targetable that in the absence, these tumors are growing slower. So then, making use of 2 pharmacological inhibitors that are available, FC9402 and HTS07545, we wanted to better understand the mechanism of what's going on here. Using in vitro systems, I used 3 parallel assays. We can see that inhibition of SQOR results in a decrease in proliferation rates and self-renewal, and this recapitulates in the other SQR inhibitor as well, although not quite as potent. And as we're targeting SQOR, we would expect that when you inhibit SQOR, that there would be an increase in accumulation of hydrogen sulfide. So we evaluated the effects of inhibiting SQR on hydrogen sulfide accumulation using a flow-based assay with a hydrogen sulfide fluorescent probe. And what we can see is that there is an increase in hydrogen sulfide accumulating in these cells with both inhibitors. And as hydrogen sulfide is accumulating, what we would expect then is that it is, first of all, not being used to generate ATP via the electron transport chain, but in fact, hydrogen sulfide, similar to like cyanide, can actually act as an inhibitor of complex 4 of the electron transport chain. So we use seahorse assay to measure oxygen consumption rate, and what we can see is that there is a decrease in oxygen consumption rate, both the basal and maximum oxygen consumption rate. So these the mitochondria are being less functional, and what we see is that there is actually this increase in extracellular acidification rate, which is indicating that these cells are becoming more glycolytic. And we also see that there is an increase in the ratio of ADP to ATP. And so then, as it appears that these cells are more glycolytic, it would be reasonable to expect that they would be more sensitive to deprivation of like glucose. So we did an experiment where we combined either the genetic knockdown or pharmacological inhibition of SQR with 2-deoxyglucose, which inhibits glycolysis. And so what we can see is that the cells are more sensitive to the combination of the absence of sulfide oxidation and an inability to perform glycolysis. But what is really interesting is that when we even go beyond just the effects on electron transport chain activity as well as glycolysis, we see that there is, as the hydrogen sulfide is accumulating, it's actually leading to an accumulation of lipids in these lipid droplets. So this occurs with both of the SQR inhibitors. And we also see an upregulation of expression of enzymes that are involved in lipid biogenesis. And we see that these cells are unable to actually oxidize lipids as a potential fuel source. And then what is actually really exciting, we did another in vivo experiment where wild-type mice were implanted just with normal cells, and then we gave a week for the tumor cells to engraft, and then we began treatment 3 times a week with the SQR inhibitor. And so what we actually see with 2 different doses, 0.5 and 1 milligram per kilogram, 3 times a week, is that we have a significant extension in survival in these mice. So this leads to the current working model that at baseline, it seems that in glioblastoma, the cells are able to perform glycolysis to meet their energetic demands, and when needed, they can perform they can utilize lipids for additional energy, and they're maintaining a constant turnover of hydrogen sulfide. Whereas when hydrogen sulfide levels increase, these cells, they upregulate the sulfide oxidation machinery, which can contribute to production of ATP while also eliminating this tumor-suppressive molecule. And then they're able to maintain their normal metabolic functions. However, when we inhibit or knock down SQR, what we see is that there is a decrease in ATP production. In fact, it looks like the cells become more glycolytic, and they're unable to break down lipids stored in lipid droplets. And so this might be a potential targetable pathway. And with that, I'd like to thank members of both the Hein Lab, the Lathia Lab, as well as all our collaborators and funding sources.
Okay. Thank you so much, Andras. Looking around, no burning questions. We'll move on. So next up is Evan Joseph, who's a PhD student in Chris Hubert's lab. And their lab was really the first to pioneer growing patient-derived tumor cells in organoids in glioblastoma, which has really set the stage for a lot of translational studies. So Evan, we look forward to your presentation.
Okay. Thank you for the introduction, Justin. I'm so excited to be here and talk about some of the stuff that we've been doing in the lab right now, revolving around stem cell maintenance and how estrogen receptor may function in sort of maintaining stem cell self-renewal, as well as how estrogen receptor may function as something that would affect the entire tumor microenvironment as a whole. So before I get right into it, Something that we love looking into in the lab right now is studying cancer stem cells in their native environment. So since there are a bunch of microenvironments in GBM, as we all know, there's a bunch of hypoxia, there's invasion along the blood vessels, and something that this IHC for SOX2 helps look at is the fact that stem cells exist as a gradient within the tumor. They like living close to the blood vessels, but as you start moving away from the blood vessels, you end up losing a bunch of stem cells. So our lab developed these organoids that have two innate microenvironments, one of which is proliferative rim, which is filled with stem cells, a lot of actively dividing, actively cycling cells, but also has its own hypoxic niche, wherein you have a lot of senescent cells, a lot of necrosis, a lot of slow cycling cells. To compare it with regular sphere culture, they're massive. They can become ginormous and huge, and they take a long time to grow. Going deeper into them, yeah, they're big. And going deeper into them, you can see that they also end up developing this sort of gradient of SOX2 that we saw in the patient tumors. And not just that, you end up seeing a bunch of hypoxia as you go deeper within the organoid, as exhibited by the CA9 marker. So something else that we can do is sort of label this outer niche of the organoid and then separate out cells from the outer niche and the inner niche of the organoid. And you can study them separately and see how they respond to treatments or drugs or stuff like that. And what the lab has already established is that when we seek out these cells from these two niches, the rim of the organoid has an expression profile that is that has a lot of overlap with the cellular tumor regions of the of actual patient tumors, so a lot of proliferation. But also the cells that are in the core of the organoid, so the hypoxic area, has an expression profile that overlaps with hypoxic cells within patient tumors as well. So what I did with this data set is figure out the top genes that happen to be upregulated in the rim of the organoid compared to the core, and performed gene set enrichment analysis and looked at the top hits. And the first thing that showed up happened to be estradiol response, which we found extremely interesting. And then we saw something called the DREAM targets. We validated these genes from the IVGAP data set as well. So before I dig into the estradiol story, just a look at the DREAM complex, something that I've been working towards quite heavily right now, because so this complex happens to be very pivotal in maintaining cell cycle. And just to take a quick look, the blue ring here is called the MUF-B complex, or the multivalvular B complex. It's called so because someone tried to mess with it a few years ago and found that if you mess with it, C. elegans end up developing a bunch of ectopic valval malformation, and that is why it's called multivalvular B complex. So definitely something's going on with sex differences and stuff like that, but also just as a base here, this MUF-B complex is very important in switching cell cycle from a repressive phase to a proliferative phase. So it can bind to a bunch of partners and form something called the DREAM complex, which would sort of suppress cell cycle, press the brakes. You'd have a lot of senescent and quiescent-like phenotype. But the same MUF-B complex, when binds to B-MIB, progresses the cell cycle, so a lot of proliferation. Something else that we looked at was how does the expression of these different members of the complex or modulators of this complex provide a survival advantage to a particular sex. And what we found was there's some sort of sex skew difference that happens. So basically what you see here is that the members of the core complex, if they're highly expressed, tend to give survival advantage only to females, but the modulators of the complex, so they don't really are a part of the complex, but can affect the activity of the complex, tends to give a survival advantage to males in particular. So some sort of sex skew difference that we are not really sure about what's happening at this point. So I started by doing some very simple experiments. I decided I would grow organoids in the presence of estradiol, as well as fulvestrant, which happens to be an estrogen receptor inhibitor. And the very first thing that we noticed, which was very clear, that fulvestrant, the estrogen receptor inhibitor, resulted in organoids that were very tiny. they happen to be this sized. You give them fulvestrant, they're growing in fulvestrant for about a month, you see the reduction in size, which is massive. And inside the organoid as well, even though it looks much prettier than the other ones, it is not what it's supposed to look like. So something's definitely happening here. So again, the next thing we would do with it is to look at the stem cells within these organoids. And what we found was growing these organoids in estradiol for a while results in a depletion in stem cells. And this happens to be true for multiple different rounds and multiple different organoids grown at different time points. And estradiol just seems to sort of repress self-renewal, but does not seem to affect physically what an organoid looks like. So putting all of these dots together has been a bit difficult, but we know that there's something about cell cycle, there's something about self-renewal. So we started by asking the question about, well, does it actually affect the cell cycle in any way? Turns out it does. So estradiol seems to have a little more cycling cells, nothing statistically significant, but fulvestrant resulted in this massive G2M block, wherein the cells were they just stopped cycling, I think. We don't know that yet. I'm assuming it's a G2M block. I am not sure. But the thing about this G2M block is that this exists in both the proliferative niche of the organoid as well as the hypoxic niche of the organoid, although it's not significant. Okay, so the next thing that I wanted to look at was how all of these treatments affected senescence. So I used a flow-based assay that measures senescence based on SA-beta-Cal, and if you look at an organoid, you would have a bunch of senescent cells usually in it. So what I saw was that the vehicle would have a certain percentage of the senescent cells, but the organoids that were grown in estradiol seemed to have a massive drop in the percent of senescent cells in them. Of course, on the contrary, organoids that were treated with fulvestrant happen to have much larger population of senescent cells in them. So what we think is happening is there's a sort of tip in the balance between the DREAM and the MMB complex, and I'm currently in the process of dissecting all of that. The next thing we wanted to look at was how does the estrogen receptor expression vary within the entire GBM tumors, in patient tumors. So we already knew that higher expression of the estrogen receptor gives a survival advantage only in males and not in females, but we wanted to know where exactly within the tumor these estrogen receptors are present. Turns out they like living close to blood vessels, and we wanted to make sure that this signature is not arising because of the endothelial cells, because they can also express estrogen receptors. Digging into the single cell-seq data from the Darwin lab, we figured out that it was malignant cells that were expressing the estrogen receptor and not the endothelial cells. So the next thing I did was look at the IVGAP database, which has about 946 tumor subblocks from 41 GBM patients, and they have areas of tumor regions quantified. So we wanted to look at how different tumor regions are different in either males or female tumors. And what we found was that female patients tend to have a lot more hyperplastic blood vessels across the tumor. So a lot more hyperplastic blood vessels, which basically means that these vessels might be leakier. And at this point, from collaborators, we have some murine data that says that female tumors may have a lot more permissive blood-brain barrier permeability, but we are in the process of looking into other models as well. So this brings me to my central hypothesis at this point, is that estrogen receptor function might be working through the MUFB complex, and this might lead to sort of driving proliferation at the expense of stem cells, but also elevated estrogen within females might provide sort of increased hyperplastic blood vessel formation as well as increased chemotherapeutic access, both of which may contribute to this increased survival in females in with GBM. And with that, I'd like to thank my lab and all of our collaborators. Justin's lab has always been so helpful. Yeah, and I'll take any questions you have.
time for one question, if there's anything. Okay, thank you. Okay, so you will see in a second why we put Evan where we did. All right. Okay. So it's a real honor to be here to present some of the work that you, Dr. Khatib, have supported. And you asked a question about, where are we going to be? Why aren't we where we're at right now? And I want to make an argument for a second that I think we've been thinking about the disease a little bit differently. And I think we have an opportunity to use inherent human biology to inform what's going on. So I want to talk about biological sex. And I want to talk about biological sex because I think it's really meaningful, but I need to first start with this diagram. You, by and large, have heard of the term gender. That's a social construct, and obviously that's on the right-hand slide. And we're very interested in biological sex. And when we started these studies about 10 years ago, I was probably one of the biggest offenders using those words interchangeably. They're completely different. So when we talk about biological sex, we think about things like hormones and chromosomes, which are really important. And I think the little known fact is actually the X chromosome has about 10 times more genes than the Y chromosome. And that really is quite important. Now, there's no doubt that sex modifies gender and gender modifies sex, but we're very interested in this concept of biological sex. And we've really been led by the epidemiology of cancer. So I like this review a lot, and really it just summarizes the fact that as a whole, males get tumors more frequently than females, and males do worse. And I'm sure, Dr. Khatib, in your own practice, when you were thinking about it, you probably operated on more men than women, right? So there's something about the biology that's really been there. And what's interesting is if you look very carefully at clinical trial outcome, you can see the same thing. This is a beautiful review from Eitan Rupin's group at NIH, where if you look at the clinical response, you look at different disease sites, and you look at different side effects, the same drugs that we're giving to males and females are not only acting differently, but having a different side effect. And I think that's actually quite important. But what's interesting is a lot of our world is modeling human disease in mice. And I was really gratified when this paper came out earlier this year, where even in mice, there's a sex difference in outcome, or rather presentation, of tumors. So mice normally don't get tumors. You let them live long enough, they do get tumors. But what's really interesting is the tumors are different between males and females. And that got us thinking about the human population, which we're very interested in treating. And this is one of my favorite diagrams. It's a bit of an older diagram. And really, it just lines individuals up by height. So 5 feet 0 inches is to the left, 6 feet 5 inches is to the right. And what's interesting is the individuals dressed in white are biological females, and the ones dressed in black are biological males. So you can see that even in this human population, if we take something as boring as height, you can still see a sex difference. So this work started really, as I said, about 10 years ago, and this was a close collaboration with Joel Barnholt Sloan, who's one of our epidemiological collaborators. We looked at patients that were treated in Ohio. So Philip, this includes Cincinnati. So we did Cincinnati, Columbus, and Cleveland in the major academic medical centers through something called a Ohio brain tumor study. What we noticed is actually that given the same standard of care therapy, right, so maximal safe surgical resection, radiation, and temozolomide, males actually had a poorer prognosis than females. And this is getting a little philosophical, but essentially the overwhelming majority of drugs were actually historically only tested on males. They were optimized for males. And even there, we're not doing very well. And what's interesting is the epidemiology of the disease suggests that if you look at all gliomas or glioblastoma, males have a higher incidence than females. So we have a tumor that manifests more frequently in males, and our quote-unquote best standard of care therapy doesn't work as well in males. It gets more complicated because there's a lot of differences in the transcriptome between males and females. And this is a study that was led by Josh Rubin that we participated in that really uncovered differences in the genetics of male and female tumors, as well as the imaging. And we took it a step further with Manmeet Ahluwalia, who's one of our neuro-oncologists who was here, who's since relocated. But one of the big clinical markers is the MGMT methylation. It provides us an opportunity to understand, will a patient actually respond to chemotherapy? So the group in blue is male. So it really doesn't matter as much if a male has MGMT methylation or not. It's actually the females where you see that huge difference. So, we're just sort of scratching the surface that some of our therapies may actually be working better in one sex than the other. And this really started a journey that we've been working on for a while. Now, I'm only going to show you diagrams and describe concepts. The majority of the work is published. So what happened is Daphne Bayek, who was in the lab, who now has her own lab at the University of Miami, and Ju-Yun Lee, who's sitting in the back of the room and is on the job market. Between the two of them, they looked very carefully at the tumor microenvironment in mouse models, but also in human patients. And what they found was in males, there's a very local immune suppression. And in females, the immune suppression is peripheral. In males, the T cells are more exhausted than females. And Ju-Yun went on to show actually that males are more responsive to immune checkpoint inhibitors. And that really dovetails with what's known in non-small cell lung cancer and melanoma. So this was just sort of a little bit more of a zoom in for her work. And what's very interesting is she uncovered something very unexpected. The reason that there was a potential sex difference was because of a process called X chromosome inactivation escape. So it turns out in mice about 6% of genes, and in humans about 20% of genes on the X chromosome undergo escape. So females have 2 copies of a gene when they should really only have 1. And when she shut that process down, she could get the female T cells to look more like male T cells. So we've taken this a step further, and we're really interested in what underlies this difference. And this is an extremely complicated story that I will not tell you, but it's publicly available, and we'll send you the paper, Dr. Khatib. So what Ju-Yun found was that androgen functions as a tumor suppressor only in the brain. So if you think about that as you get older, males in particular, the level of androgens is reduced, and it turns out that male incidence, and actually even female incidence of glioblastoma is around 65 years of age. And what's interesting about this study is that upon the loss of androgen, the HPA axis gets hyperactive. And what ends up happening is that generates a lot of glucocorticoids, which drives immune suppression and ineffective tumor control. So in the age of cancer neuroscience, when we're thinking about how tumors are interacting and talking with neurons, there's actually hardwired physiological systems that are also being impacted by the tumor growing in there. And we've done some experiments where she's even put in beads, fluorescent beads, and we can evoke the same hormonal response. So that's the story about testosterone and androgens. And another story that we're just finishing up here, led by Anthony, who's sitting in the back, actually just to the left of Ju-Yun, involves platelets. So we've been very interested in this idea of platelets in cancer. It turns out that the second leading death of cancer patients is actually thrombotic events. So what Anthony's been able to find is that there's actually drugs that we can use to inhibit this process. They only work in females, which is quite interesting for us to think about. And what he's done is he's been able to show a very elegant interaction between estrogen signaling and the PAR4 receptor that underlie that. So again, this is just another example of how, as a human population, you've got these differences, and are they actually actionable? Can we use them to our advantage for therapeutic development? And that really brings me to this idea of an integrated approach. We're interested in a lot of aspects of biology, but what I would argue is at the top of a lot of these, and what shapes a lot of these, are things like hormones and chromosomes, and there are ways to manipulate them. And obviously they're going to have downstream consequences, because I've given you examples where there's a sex difference in the response to a lot of things that we're giving patients right now. So I think, when we think about precision medicine, I think we have to think about a human population a little bit differently. So I would argue that there are sex differences. Some of them are actually drivers of disease, certainly accelerators. Drug responses show sex differences. When we first started this work, people were a little bit confused. They said, we don't believe this. And then I show them this drug called Ambien. Ambien actually has a different dosing, whether you're a male or a female, which is actually pretty important. So I think we're at a place now where we actually need to be thinking about this, not only for our human studies, but also in our mouse models. So I want to close with a series of thank yous. First is to thank you, Dr. Khatib, for all of your support and inspiration. So obviously, it's a great photo of us at dinner with Sina in Florida last year. We really appreciated your time and letting us talk to you about some of the work we're doing. I'd be remiss if I didn't thank the laboratory. We've had a really good run over the last couple of years. The folks in yellow are still in the lab, and the folks in pink have gone off and started their own labs, so we're building quite a team. We've got great collaborators here within town, throughout the US and the world, who've really helped with this. And then I wanted to end with just thanking the program in general at the Case Comprehensive Cancer Center, because as Dr. Schwartz said, it's a real team environment. And I think his statements were ambitious to suggest that the Guardians, the Browns, and the Cavs would all have great years at the same time. But I think in neuro-oncology we are. And I tell people how big our program is, and no one really believes me. So these are the people who run labs at the Cancer Center, who are at the Cleveland Clinic. And these are fully funded brain tumor folks. And you can see a lot of them are in the room. A lot of them were represented. At Case Western and University Hospitals, we have a similar team. We also have done a really good job within the city of convincing really talented people who don't necessarily think about brain tumors to start studying brain tumors. And you heard talks from several of those labs today. And that extends not only at Cleveland Clinic, but also at Cleveland State, which is a very strong engineering school down the street. So I'm going to stop there. And these are the folks in the lab jumping for joy. And I want to thank you again, Dr. Khatib. And I know we're running short on time. So what I'm going to do is actually turn it over to Dr. Mohammadi, who is going to present some of his recent work on some exciting translational science. So thanks again.
All right. Thank you very much. So great talks today, and most of my colleagues, like great scientists, they showed what they do in their lab. I'm going to just approach it from a different way. Like, I'm a fully clinical neurosurgeon, but I'm just like a phone call away from all of them. I collaborate. We have been collaborating a lot with the labs in the Lerner Research Institute, and I'm going to just touch two of the studies that, as a matter of fact, we used Dr. Khatib fund for them. So, the first study, initially sponsored by the NICO, it was a company that doesn't exist actually, bought by Stryker, I guess. And we had to use Dr. Khatib fund for the additional analysis. And the second study was the one that actually Jennifer Yu presented. So I'm going to just touch very shortly on that one too. So one thing that, for surgical trials that I'm involved in, one thing that is very important for us, we don't delay the surgery for a day to enroll a patient in a trial. Many of our patients, they are coming through hospital transfer from our satellites for surgery, and the surgery, is scheduled based on OR availability in whatever the first available OR that I operate twice a week on Wednesday, Fridays. And when the surgery is scheduled, then I look to see if it's eligible for any trial. Many of them are in a very short notice, and these trials, they need multiple disciplines to be involved, including from the clinical people to lab to research nurses, and somehow it always happens. The first study that I collaborated with Justin Lab, we call it for short NICO study, and the second that I'm doing it with Jennifer's lab is recording study. These are the two surgical trials that we use the newly diagnosed GBM for, like collaboration with the lab. So the NICO study, the idea was, as you can see in this, when we do surgery for GBM, like we have intraoperative MRI, so we do intraop MRI at the middle. So initially, you remove the core of the tumor. When you get the intraop MRI, you may or may not have a little bit residual at the enhancing rim, and you go back and remove that. What you do radiation and temozolomide is the non-enhancing portion, the like leading edge of the tumor or infiltrative part of the tumor. So the idea was to see if we are treating the right thing, because the information that we have is mostly from the core of the tumor, a little bit from the rim, but we are treating the infiltrative non-enhancing tumor. So in this study, we planned 3 trajectories, one going to the non-enhancing portion of the tumor, one to the enhancing rim, and the other one to the central core. And we got 3 samples and send it to the Justin Labs, as I will let you know. So this is the NICO device that is like a cutting tool with a suction that we would use the navigation at the end of this device so we could track the tip where it goes, and we go to each trajectory and collect the tissue. And this is a tissue preserver, which is like a suction trap, and it traps all of the tissue that we get from each of the trajectories. And it does have this container for ice water, or we put just ice there. And when we collect the tissue, then this way, and you get plenty of tissue with that device. We put it in the ice cold container, and the same day we would send this to Justin's lab to do the initial steps for the preparing the tissue, and then we have this amount of tissue that we are using it. I think the study finished like 2 years ago, and we're still just running new tests. The last one, actually, Andrew is working on it, is single-cell RNA sequencing. So, and when the tissue goes to the lab, so I think I'm going to skip the technical part. That's what Justin and the lab were doing, and this is happening the same day. It's like about 45 minutes from the time that I remove the tissue. It goes directly to the lab, and they start processing right away. And these are the tests that we have done so far. The flow cytometry, the first 2 was done by Justin's lab, and the MRI sequencing, we are doing it in other. We enrolled 20 patients in this newly diagnosed GBM. And so that's the thing. So when the patient comes, it needs a lot of steps for the patient until it gets to surgery. But this is happening in a couple days. So it was like a group email that goes to Justin's lab, to our research nurses. For this study, we had a representative from the company coming for each case to help with the that tissue preservation. It was not commercially available at the time. And and everybody and the research nurses, they evaluate the patient. If it's good to go, they fill the forms, put it on my desk. Many times I had to sign it like the morning of surgery before I go to the OR. And then we collect the tissue, And in 45 minutes, that was in Justin's labs that they were standby for our surgery, and they would the processing. They would do the processing. And this is how it looks like. This is intraoperative view. And this is the NICO Myriad device with the brain lab, the navigation star at the end of it. And what happens is here you can see in the navigation system, we can track the tip where it goes. And we go one by one on each trajectory, collect the tissue, flush the NICO and then go to the second trajectory and the third trajectory. With this, we had like 60 samples, like 3 for each patient. And this is another picture. So this is our, I think it must be our first patient. That this is the screenshots, intraoperative screenshots for each of the trajectories. This is the non-enhancing, rim enhancing, and central core. And after we did that, then we removed the rest of the tumor. So with putting the tissue in an ice-cold container and sending in like 45 minutes to Justin's lab and start processing, more than 80% of the tissue was still viable by the time that they were in the lab. And these are like some results of the test that this is the immunoprofiling that was done in Justin's lab. And it shows that in non-enhancing tumor, we have lower frequency of the infiltrative immune cells compared to the enhancing. I mean, you would think that would be one of the reasons that immunotherapy doesn't work, because we always remove the enhancing portion and leave the non-enhancing with like fewer immune cells. And the functionality of the T cell was the same. The bulk RNA sequencing showed some differences, in the enhancing portion and the non-enhancing. So the non-enhancing portion was more the characteristics of the brain. And so this part is what we are doing in the second study, the recording study with Jennifer, just doing single-cell RNA sequencing in different areas. And this is the immunoprofiling that for all 20 patients that was done in Justin lab. For a whole exome sequencing, although the tumor mutational burden was uniform across the regions, but there are some differences, like this is the KMT2C, which is different in the non-enhancing portion versus the tumor. So there are some differences. And the single-cell RNA sequencing, actually Andrew is working on it. He's like the expert on this, as like a PhD from Oxford for that. And hopefully we are presenting it in SNO meeting this fall. The second study, so Jennifer explained very nicely. I'm going to just approach from the different way. So the depth recording, although it's been used in Europe, but brought to the US by Cleveland Clinic, by Jorge Gonzalez Martinez, one of our epilepsy surgeons, like 15 years ago. And it's used very frequently for the epilepsy surgeries to detect the deep-seated epilepsy centers. And our epilepsy team, the neuroepileptologists and the epilepsy surgeons are very expert on using this. So it was very easy for us to just include them in our, the team. And, although then Jen mentioned this study, and they had, some idea about the tumor-neuron interaction, but in the human level, nobody has done the depth recording. It was just a superficial grid, which makes it less effective. But one thing that I was going to mention here, so this study was a little bit more complicated because more people were involved. So Jennifer used lab. So I was doing the surgery. This is our neuroradiologist. This is the Juan Belaja is from the epilepsy neuroepileptologist, and Rafia Vetzian is from the neuroanesthesiologist, because we have to use a specific type of anesthesiology that they use for depth recording in epilepsy people. Actually, we had to change it after the first case because the epilepsy people tend to be younger. Our patients, they were older, so we have to adjust it based on the age. But again, when we had a patient, the email would go to like a larger group to be ready for the day of surgery. And this, Jennifer showed this too, but we would plan like in 4 different areas anatomically and 1 in the center. And this is and all of the electrodes are marked by location and ABCD, and that's how we did it, the recording. So we do 10 minutes of recording before starting the resection of tumor, and they keep this recording until progression happens, and then based on which direction we have the progression, they go back and analyze the recording. They segment that 10 minutes, which is 600 seconds, to 10-second segments. So then they can use whatever, like AI or whatever the tools that they have to see if there is any differences between the electrodes that they had, the sites that they had progression versus those they don't. And the good thing of this is we have our control embedded in the, so like let's say if D and A, they have progression, C and B that don't have progression would be our control. And again, so this is one of the electrodes that had progression. This is the pre-op, which the electrode is located in the non-enhancing portion of the tumor. This is the post-op, immediate post-op, and this is the progression. The progression is a little bit challenging because the surgical cavity shrinks after the surgery. So we have to get our neuroradiology involved to, first of all, based on the perfusion and the enhancement, define where is the progression. Then we import all of those scans to the brain lab and check with each electrode to see where we have the progression. And when we let the epileptologist about the progression, for example, in this case, the superior had the way that we call it 2+ progression, and the inferior had 1+ progression. Then they do the analysis based on 10-second segments. So this study, we are going to enroll 10 patients. We already had 6. 1 of them had progression so far that I showed you. But, this is our team, but none of this would have happened without your generous donation. Dr. Khatib is like extremely helpful, especially for these early surgical trials. And although this study, we're going to apply for the SPORE with Justin and others to get like more funding, hopefully. But to explore new ideas, I think that's tremendously helpful. Thank you very much.
Okay, thank you. So that's actually the end of the presentations. I guess it's your turn, Dr. Khatib. Would you like to say a few words? We have a recording as well from you.
Probably. I would like to thank everyone who is here, and also I have something to that said on it.
Okay.
Yeah.
Okay, so we'll go ahead and play the recording.
Ladies and gentlemen, I thank Case Comprehensive Cancer Center for hosting this symposium. Thank you, Dean Gerson and Dr. Nadia, and a special thanks to Dr. Sina Ahmadi for organizing this event. I knew a musician who had a wonderful career as a piano soloist. And when the age made his own traveling and performing difficult, he became a successful orchestral conductor. His purpose in life had grown behind making music himself to encouraging and supporting others. My profession has been medicine, and my most important fight against GBM and other brain illness. So in retirement, my purpose became to have the fruit of my career be a tool for supporting and helping as you continue this important fight. I am honored by and grateful for the music you are making. The Khatib Foundation story began 80 years ago in the holy city of Basha. As a boy of 10, I learned from my parents that a greatly admired public official had suffered an epidural hematoma. It claimed his life. That started me on my path to Tehran Medical School to neurosurgery at the United States. For 60 years, I had been fighting the war against glioblastoma multiforme and other brain illness. As a practicing surgeon, I repeatedly felt pain, anguish, and frustration of patients and of colleagues as GBM claimed life and families. My wife Georgiana and I initiated the Khatib Foundation that will support in perpetuity some of our profession's best and brightest working to end GBM. Georgiana passed away 3 years ago. I often recall her tremendous capacity for empathy and her ability to relate to people of many faiths. So it is especially gratifying to see the international and multidisciplinary body of professionals that have brought many talents to the fight. I have no doubt you and those who follow will put an end to GBM. The Riza Khatib MD Brain Tumor Research Center at Johns Hopkins to their credit. I thank Dr. Henry Brain of Johns Hopkins, a valued Khatib Foundation trustee, also to Dr. Shalman Shah, a foundation trustee. Her late husband, Dr. Andy Parsa, had honored us as the foundation's first endowed chairperson at the University of California. Join me in remembering the departed souls who may have guided you. They remain with us. Most of us owe much of who we are to who they were.
Okay, well, thank you for that great inspirational message, Dr. Khatib. So, before we close out, we just wanted to present you a small token of our appreciation. As you put it, you are our maestro, and this is the least we could do. So, Sina, do you want to come up with me? So this is on behalf of the Case Comprehensive Cancer Center. It's really our honor to present you this token of our appreciation. So, okay.
Thank you. Thank you. Thank you. That's the only thing. Just thank you very much.
Thank you.
Good.
Well, thank you. Okay, so this officially closes out. So one quick announcement for those who spoke at the symposium. If we could meet in the lobby for a photo, we'd appreciate it. So thanks again for coming. Hopefully it was an informative day. And as Dr. Khatib had suggested, please continue to work harder to find a cure for glioblastoma. So thanks again.